marks the beginning of the 300-meter depth transect, UTC 1326 mark.
Terrific. So we are going.
Start transect 1.
Yep, you can see it. That's great.
Happily.
Siphonophore upper right. Hopefully we'll get a good view of one of these siphonophores, which seems to be pretty common in the at this depth so far.
So is it better to drive into the current or like crab? We're facing into the current. Okay. Mm-hmm. Okay. So a lot of this is just marine snow, right, where it's not really moving?
I believe it is marine snow, yeah. That's just detritus and mucus accumulated. It's in the upper left there. Yeah, not sure what it's getting towards center, yeah. There we go. That's a nice little siphonophore. So these are modular organisms.
Cool.
Right. Uh-huh. I'm sure we'll... Oh, another little siphonophore floating by.
There's a little flat guy.
That's Keatingnath or a fish, yeah.
Oh, Medusa. Oh, I went past.
Oh. Good eyes.
Yes.
Yep. Understood.
Warp speed, Star Wars.
And then when you're out there, we can then focus on things. Great.
That might have been
That was a Medusa top right, yeah. Would have been a...
Here we go, Keatingnath at 3 o'clock, 2 o'clock, and now it's center screen right there. Little white guy. I mean, that narrows it down, right?
Yeah.
That clear thing. Oh, there it goes.
Oh, it
took off.
They're very fast when they want to be.
Mm-hmm.
Yeah, I think that's a good approach.
Here we go, some more. What's this?
Take a peek at whatever this is.
Ooh.
Oh, nice.
Cool.
What is it?
Gelatinous thing. Actinophore, maybe?
No, I don't. Do you see any of the combs?
Mm-mm. That's going to be my default all day. If I don't know what it is, it's a good actinophore.
That's all right.
Oh, neat.
Oh, I think that might have been a pteropod.
Yeah, it looks like a glass house, doesn't it?
Yes.
What's that?
It's a glass house, right?
Here's another Keatingnath, maybe?
Yeah.
Hey, Alan, Mike here.
Hey, Mike.
I was thinking maybe pteropod, but then when it got up close to it, it didn't look like any pteropod I'm familiar with. I thought it might have been a detached bract from a siphonophore.
Siphonophore.
So a loose piece of a siphonophore.
Yeah, that's my guess at the moment.
I trust you.
Just a wild guess.
All right.
I got something moving at 3 o'clock, real small.
Yeah.
I have no
It's
idea what
going
that
up
is.
high. It's not in focus.
Yeah.
It's definitely challenging to get things in focus, but we'll find some things that behave.
Left is center. There we go. The Okeanos maybe. It's just out of focus. Yeah, now it's at 9. Yeah.
Oh.
Oh, there goes the. It might have just been schmutz. Jelly goo. Another Okeanos, I think.
Tied up nutrients, you mean?
Yeah, schmutz.
Schmutz.
Here's a.
Everyone on shore to stand by. We're going to do a pilot change. It takes a lot of focus and energy to pilot through the water column, so that's why we're not moving as quick right now.
You know, while they're doing the pilot change, it's worth mentioning to people that are watching ashore that this is the shallowest transect, and during this time of day when the sun's out, a lot of the animals have gone deeper into the water column. So, this is where I would expect to see the least stuff. And then as we get down deeper, particularly to the deep scattering layer, there's going to be a lot more encounters per minute or whatever.
There's a Medusa in the top.
At 1 o'clock off screen.
Yeah, thanks for mentioning that, Mike.
Yeah, so we expect not to be too much here. Go slow and zoom in on what we can.
One of our transects will be in the middle of the deep scattering layer, which is the layer that should have the densest concentration of midwater organisms that we will see. That was fast.
I'm seeing these little white, they have more substance to them. Like little white beady balls, I guess. I don't know what they are. Maybe eggs or detritus. Yeah, they just,
Yeah,
they have like they're a little more like, they're less clear. There's something. There's something. Ooh.
Yeah. Oh, top right.
Oh, 6 o'clock below screen. Or right there, like a ring. It's coming into focus now.
Good eyes, Steph.
Yeah.
Yeah, I think that.
Can you zoom on that or no? It might be out of. It's between our knees.
Don't see it pulsing like a Medusa, but the ring made me think that it might be.
Hey, this is Matt. I just want to tell everyone on shore We have changed the way we transmit our audio to the streams. So it's kind of an experiment this cruise, but stream 1 will have science audio, stream 2 will have the ROV pilot audio, and stream 3 is going to have both mixed together. So if you want to hear the ROV pilot's input, check out one of the other 2 streams besides stream 1, and you can hear them. But stream 3 has both, so that's the best of both worlds right there. Thank you.
Stream 3 also has the our depth monitoring.
Keating Nath.
There we go. Something at noon. Okay.
I changed the quad split to include the ROV and CPHD cameras on the bottom.
I then.
I was not hearing pilots, Mike.
There we see a siphonophore in the top.
Here we go. At noon, off noon, and what noon? Here's another one. Here's something.
I think that's a little bit of
Schmutz.
marine snow. Schmutz, yes.
We got something flashing thing at 3 o'clock, it looks like. I think I might be able to see it anymore.
There's something swimming across to the left. That had the wavy up and down like it might have been a pteropod.
Mm-hmm.
Ooh, left. Ooh.
There we go. Okay.
Gotcha.
So we're going to speed up, get the end of the tether, and then we'll be able to focus again.
Definitely difficult piloting work.
Here's something neat.
Something swimming.
Whoop. Here's something at 3. Here we go. Now it's at noon. Looks like a siphonophore, maybe?
Definitely a siphonophore.
Look at it spinning around.
You can see it's got its stem
Ooh.
deployed, and there it is. It's going to race away.
Neat. There's another one at noon.
These animals are sit and wait predators, so they will they're basically a colony, lots of different individuals that are clones of each other, but are very different in appearance. So some of them are specialized for swimming, others of the individuals in this colony are specialized for killing food and eating it, and still others for reproduction. And they will deploy their tentacles out in the water column until organisms contact it. And then, of course, we've seen their quick escape responses to D2 pushing through the water.
Oops, something at 9. There, now it's center screen. Now it's at 3. There you go. It's hard to get a focus on.
Yeah.
These flashy things, I'm not sure what they are. I'm seeing it. Sometimes they're ketognaths, like clear.
That reflect back the lights. Just joining us, this is our first transect through the Midwater column today. It's at 300 meters, and this is the depth at which we expect the lowest density of organisms, because it is daylight out today, and many of the zooplankters have migrated to deeper, darker waters where it's easier to not be detected by visual predators.
Most of the stuff here seems to be just clear. It's all clear. I don't see any reds. Sometimes, you see the jellyfish at the red.
I'm sure before
guts,
the day's over, we're going to see some red guts.
Here looks like another siphonophore at 9 o'clock.
Siphonophore all deployed.
Ooh.
Oh, and it's going to run away from us as well. Oh, there goes the siphonophore. So like a lot of midwater organisms, the siphonophores are very specialized for whatever prey they prefer. So some are specialists on small crustaceans, others on large crustaceans, still others, off to the right, will eat fishes. The ones that was a nice view of a ketognath that came by on the left. And they have
here's another siphonophore at, I think, at 6.
Okay.
Here we go, 3 o'clock.
Another nice siphonophore. So I was saying they have morphologies that reflect their specific prey items. So a lot of times the tentacles, they have little side branches called tentilla, and some of those can be have been modified as to be lures, as luminescent or fluorescent lures.
Mm-hmm. Oh, got a jelly at noon off screen.
Maybe a larvation house. I don't know. It went by kind of fast. I heard that.
Like, oh. So the little siphonophores have those little white, they have the white, like, beady balls, right? Little white spots.
Yes.
It looks like a lot of these might be. Could they be broken off pieces of those? They're the same texture.
They might be. Siphonophores will actually break off. Nice.
Yeah, see what I'm, like these little white.
Yeah.
Mm-hmm.
It's all right. It's a challenge. There's another one straight ahead. These are pretty small siphonophores. Some of them can be many, many meters long, but I assume that these are quite small.
Mm-hmm.
Maybe the species
There you go.
Nanomia. This is really
Nanomia?
Nanomia is a small siphonophore it's very abundant in the So we're pushing out through this 300 meters again. And we can slow down. Ooh, bottom left.
Something at 8.
Whoa, that...
Just off screen. Oh. There it is. Ah, that looked similar to the one we saw earlier.
It did. Again, not swimming, but...
Mm-hmm. Circular.
Yeah.
It's difficult to focus.
Yeah.
You know, until it's like there's nothing really to focus on until it's too late. And it's past
sit in
us.
my chair.
Yeah, I should sit in Roland's chair. Roland's trying to focus on all this stuff.
We'll get some...
There's my
a
Keating now.
Keating Nath, an arrowworm. It's a predator with really amazing jaws.
Aren't they a chordate? They're chordate, correct?
No.
They're not a chordate?
No. They are just their own phylum out there in the middle of the animal kingdom. Their phylogenetic position has actually been very difficult to nail down.
Oh, here we go. Bye.
It's just so
it's gone.
fast.
I know, they need to sit still.
It's eat or be eaten out here.
Yeah, for real.
So there are lots of organisms that are making themselves not easy to see from the organisms that have eyes, and then a lot of them have quite nice escape swimming behaviors. Some of those siphonophores are really fast.
I'll also mention that I've done a lot of these types of dives, and it's been my impression that when you get below about 500 meters, the behavior, the reaction of the animals to the sub and its lights, it changes. So when you're up here at like 300 meters, they run away real fast. Mm-hmm. But then when you get deeper and it's darker, they more freeze in place, and you have a better chance of getting a close-up of them. And I think that's because once you get down into the darker water, the lights of the sub are something that they don't know how to deal with. It's something beyond their evolutionary experience, so they just don't have a quick reaction to it.
Huh.
When we're in the deep sea, the water movement can also give away an animal's position too. So I wonder if freezing still would prevent that. You know, you're no longer.
It's one way of not being sensed.
You're right, because you're not moving the water around you.
So all the squids on this transect are disappearing just before we get there.
I hope so.
They're probably behind us, though.
Behind us, yeah.
With 2 fingers up.
Ketic Nath at 6.
Mm-hmm.
Not moving. Ooh.
It's good to know to get the most difficult transect out of the way first.
Yeah. Here it looks like a shrimp, maybe? What's that? Oh, 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 oh.
Oh, yeah.
Maybe a copepod?
I think it's a shrimp. Oh, well.
Bye-bye.
Now that was probably good enough video for a crustacean-oriented person to know exactly what it was. I hope.
Yeah.
All right, pushing out again.
Oh, here's a Medusa at noon.
Oh, yeah.
Coming right into view.
Just a little bit high.
Yeah. And there it goes.
That was a Narcomedusa solmissus.
The further we look up, the more light that comes and hits the housing on the camera. So it makes it a little more difficult to see.
The contrast goes down.
Mm-hmm.
Yeah, that the direction of the light plays a role in a lot of adaptations that these mesopelagic organisms have. Some of them will be, they will, because looking up and looking down are different, so the tops and the bottoms of the organisms can be different. Some fishes will do this counter illumination where they, squids too.
They light up their underside.
Yeah, so that
the lanternfish do that, don't
so
they?
that they match.
Sorry, Mike, what?
The shrimps do it, too.
Shrimps.
Yeah, I believe I saw a talk about shrimps and their counter illumination. They also have, like sort of eyes on the sides of their bodies to sense the surrounding. Oh, there was a Medusa below us.
There's something at noon, a little white blob. Just out of focus there. Oh, there we go. So below screen now. We got something at 3. Looks like hair.
Ooh.
There you go. Yep.
This would be great to get some video on this. No, I believe that was I think it just went by.
Foraminifera?
Yeah, or radiolarian, some sort of large protist with extended pseudopods. So it's also another predatory organism, but just a single cell.
It's obvious from listening to your narrative that you have a much clearer picture than I do here on the low latency internet feed, because you're seeing things that I may or may not see. But like for that protist,
yeah,
I saw a little bit of the pseudopods, and I agree that that's probably what it was, but I couldn't see enough to tell anything about it.
Oh, here's something. There we go.
There we have a little, that's a halicreatid trachea. Oh, no, it's not. It's just a bit of, it's marine snow. Mucus, old discarded mucus house maybe of a larvacean. So, snot.
Snot, sea snot.
I was hoping it was and that's probably what we saw before that was sort of looking a bit like a medusa with a fringe. But the
pteropods create mucus webs also for feeding.
Oh,
and their mucus webs are not as structured as the ones from larvaceans. That might have been one from a pteropod.
thanks, Mike. I would like to get some good footage of pteropods on this before the end of the day.
Looks like a siphonophore at 6, just below screen, or just coming. Oh, there's another one at noon. And that one's off. Yeah. We have a medusa here at 9. Maybe. Maybe it's just sea snot.
Good eyes, Steph.
There you go. Now that looks different than what we saw before, huh? Ooh, it's got maybe a ctenophore. It looks like maybe some cones.
I do see the refracting light. I think that is a ctenophore. And it looks like a really highly modified lobate called Thalassocalece. It's actually has sort of a Medusa-like body form.
Oh, another one right here?
Yeah, I think that's the same one. That's the same one. We've been watching.
So now it's center screen. There we go. Cool.
Yeah, you can see the refracting comb rows on this ctenophore.
Ooh.
Oh, it's going for a little, yeah. So that, if I was right about what kind of ctenophore that is, it's it basically has a Medusa-like body form, and it actually encloses prey inside the space, and then the prey gets confused until it wanders into its mouth and gets sort of ingested that way. It's a very, very delicate organism.
seeing mostly Cadignaths. There goes one. And Siphonophores, it looks like they're the dominant fauna type. All right, we've got 5 minutes left in the 300-meter transect, and then we're going to drop down to, what, 500 is next?
500 will be next.
500 and do another water sample, eDNA sample.
Yeah, and so, we'll have the eDNA sample along with this transect, and we'll be able to, see how those eDNA samples differ from depth to depth, as well as how our annotations of what organisms we see through the video cameras.
Here's a Cadignath. We might be able to focus on it at 10 o'clock.
Top right.
There's a Medusa, top right. Now he's center screen.
Yep. You got him. Okay. Well,
for the eDNA, it'll also be interesting to see whether the vertical migrators leave a trace in the shallower waters as they move through into the deeper waters.
Uh-huh.
So there could be some showing up in the eDNA that passed through here but are now deeper.
Oh, neat. Look how pretty that is. It's like reflects purple and green. So what do you call this guy? What's his name?
I'm not actually sure the name of this guy. What I'm looking for is the radial canals and whether there are gonads on the radial canals. This looks a bit like Tetrorchus, but usually you can see 4 gonads more clearly, but it's a similar shape Medusa with a small number of tentacles.
This is a hydroid Medusa?
This would be, if it were Tetrorchus, it would be a Tracheomedusa. So yes, it's a kind of Hydromedusa. It's definitely a Hydromedusa. So it's not in the group Scyphozoa or Cubozoa. That's a hydrozoan. And that was nice footage of that Medusa.
Mm-hmm. There's another Cadignath.
Yep. You're good with your clock direction stuff. You're really fast with that.
I'm terrible at reading analog clocks because my dyslexia messes up the arms. Like I'm okay with the hour.
That's okay. Who's that little flapping guy? That might be a That might be a pteropod.
Or a sea butterfly.
Almost. Yeah, but I think that was a pteropod, based on the way.
I agree on that. It's flying just like a pteropod. One of the thecosomes.
One of the thecosomes?
Yeah, the shelled pteropod.
Yep. Its central mass was pretty, beefy. Sort of more like Cavallina type.
Here's another Cadignath at 9. Nope, maybe not.
No,
Siphonophore?
I think that's a siphonophore, yeah.
Oh, look at him spinning around. That's neat.
Oh, yeah, you can see. all its tentacles are deployed, so you can see how it puts out a great big large net to catch prey, and now it's oh, there it goes. It's got
neat.
two. I know what that is. I think this is end of the transect, right there with our nice siphonophore. That is a calicopheran siphonophore. And I think it was desmophyce.
I'm going to need you to check my spelling on these.
Yeah, that's all right. Also, that's a little bit of a guessing beyond what we should, but
Yeah.
but it's it had two large swimming bells out front of
neat.
it that were sort of bulbous shaped. So
we're ending Transect 1.
that
was our 300
40
meters.
minutes at 300 meters.
We're going to descend to 500 meters and start Transect 2. Well, get a water sample and then start Transect 2. Oh. We got something center screen. It's flashing, and it's gone. All right, so our pilots are just setting up to make a move.
Hey, Steph.
Hey, what's up?
What was your favorite thing so far?
Well, that siphonophore we just saw was pretty cool.
Oh man, I missed that one.
Yeah, he was like flapping in the breeze.
That's cool.
There wasn't much in that column, in that 300 meter column. We'll probably see more when we get deeper.
I guess Mike had the explanation for that already, huh? It's like during the daytime, everything wants to go deeper.
Mm-hmm. Got something, maybe a pteropod shell or something at 3.
So if you're just popping in, this is NOAA Ocean Exploration EX2107 Dive 3, which we've dubbed Midwater North, and we are out on the edge of the Blake Plateau in about 1,080 meters of water, and we are doing a series of transects through the mesopelagic zone at 300 meters, which we have completed, and we're moving down to 500 meters, and after that we will be doing a transect through the deep scattering layer, which is the layer of the ocean where there's the greatest density of zooplankton, and that's at about 600 meters, and then down from there to 700, 900, and the final transect of the day is going to be 10 meters above the ocean floor.
Yeah, awesome. All right, we're descending now because we were holding at 300 for a bit there, so we're going to start descending. We're at 310. Just give us a few more minutes and we'll be ready for Transect 2.
Yep, and just before we start Transect 2 at 500 meters, we will be making our second water collection of the day for environmental DNA.
You'll see the screen get darker likely. The surrounding ambient light will start to disappear and dissipate a little more. We may not be able to see it though because we have so many lights. But
And it should be easier to see the midwater organisms on the next transect. Hopefully.
you never know what you're going to see.
Exactly.
Did you decide which transect is not going to get an eDNA sample?
Yes, I think we're going to skip the 700 meter one. So we'll do it at 300, 500, then make sure we get one in the deep scattering layer. And since that's close to 700, we'll not do 700. And then we'll do the last two eDNA samples associated with 900 and then 10 meters above the bottom. Sound good, Mike?
Just curious. Yeah, it did.
All right, 350 meters.
So we're still descending to 500 meters for our second transect, going past about 380 meters now.
We're hitting, let's see, 400 meters about. So we passed 400 meters there. So this biome, the midwater, the water column biome, is often described as the least explored of all that. Mm-hmm.
And part of it is its immensity, but also the other part of it is just that it's always moving. You know, you can't go back to the exact same spot. Well, you can go back to the exact same spot, but it's not going to be the same when you go back there. Whereas if you go down to a Lophelia mound, you can go back to that same Lophelia mound later.
And get time comparisons.
Yeah.
Plus, we've just recently, I mean, if you're looking at, you know, in scientific terms, just recently got access to this level of the ocean, you know.
Yes.
Just been able to break the surface and videotape and study it in the past 100 years or so. If that, even that long. 50 maybe.
Yeah. I would say that it seems like the pace of exploration of midwater has really picked up in recent decades.
And better access to it.
Much better access. And seeing organisms in new ways, whereas they used to always be net caught.
Yeah, and you know, these gelatinous midwater critters don't make it in the net. You know?
Some don't make it at all, and then often they'll be damaged. And one thing you also lose is the ability to, you know, we get to see behaviors, and that's something that you can't see in a net, really. Even when you have an organism in a net and it's got something else in its mouth or stomach, you don't know if it was caught in the net and it was sort of a natural occurrence, or whether it was just something happenstance.
Yeah. We're at 485.
Well, another thing these
Go ahead, Mike.
another thing these insight observations have allowed us to see is that it turns out that these animals are often in very narrow depth bands, and when you're towing a net, even for the kind that you can open and close at different depths, you're sampling over a few hundred meters, and you're not going to pick up the fact that all the animals in a species are found within a 5 or 10 meter thick depth band that's very specific. Sampling in 3 dimensions is much more difficult than the 2 dimensions when you're sampling bottom ecosystems.
Yeah, definitely. And we were just struggling to focus on stuff. All right, we've hit 500 meters. We're going to settle in, take our eDNA sample. What is this guy? A little fish, maybe?
I think that's a fish. Might be a cyclothony. Bristlemouth. So
I think you were right, Alan. Just as it drifted off to the side, it looked like a cyclothony.
That's got to be a first. Alan got a fish right. Backbone.
Well, maybe we should mention the cyclothony is the most abundant vertebrate on the Earth. That, even though you don't see them schooling together in dense aggregations or anything like that, because there's so much volume in the deep sea, even being spread out the way there are, there's lots and lots and lots of these little bristlemouth fishes called cyclothony. And if you do the math, it turns out that there's many, many more of them than any vertebrate on the planet. And most people think of the abundant vertebrates as being things like wildebeest in Africa and that sort of thing. But it's these little fishes in the deep sea that are really abundant.
So, something like, I guess the estimate is a quadrillion individuals, which is a number
We can't.
beyond our comprehension. So we're lining things up for our second transect.
Then sample.
And thank you. Success?
Did it go? So that was sample 2.
Great.
So sample 2 water. All right. Are we starting Transect 2?
And now we've got a fish in the water. Maybe another. Yes.
Yes.
Correct. Pesky Niskin 2.
Mm-hmm. Okay, that's official start of Transect 2.
Great. Transect 2 has begun and will continue for 40 minutes at this depth of 500 meters.
Oh, here's one of those bristle mouths, you think, yeah?
I think so.
We can zoom in on this little fish.
These guys have little tiny eyes and a very large mouth.
He's so tiny. They're not very graceful swimmers. They're kind of goofy.
They look kind of weak, right?
Mm-hmm.
Yeah, I agree. That's probably a cyclothone. And there's a couple of species. There are not very many species globally, but in these waters there are a couple that takes an expert to get a good look at them to tell them apart.
Great. So if we get good good imagery of them, they can be identified as species?
Yeah, I think somebody like Tracy Sutton could be able to tell you.
Nice. Okay.
Looks like another one. They
believe-
kind of like scoot forward and stop, scoot forward and stop.
That one's got a little more oomph to them.
Yeah.
We'll get a nice... Oh, almost.
Here's something Medusa or something hairy at
Ooh,
2.
Yeah.
It's coming into view right there.
Ooh, it just raised its tentacles. It's
Yep, off screen. Yep, there it goes. I don't know what that was. Any idea?
I think it was a Narcomedusa. Definitely. Ooh.
All right, so we're going to speed forward, stretch out the umbilical cord between D2 and Sirius, and then makes it a little easier for us to focus on things as we drift back. So it looks like
I think there's a lot of them in the water,
Maybe
whatever we saw before,
yeah,
so we
it's
should be able to get
Medusa.
a good view of it.
Yeah, it's already clear there's more stuff at this depth than there was at the first transect.
Yep.
We've got, here we go at 3 o'clock. There we go.
Wow. Those were, these look like little Phaedarians.
Looks like
Maybe.
similar thing at noon.
Right. I think this is a protist.
Here's another one at 2. There it goes. Oh, it's off screen. There's a
Take a
bristlemouth.
look at the bristlemouth.
There you go.
Nice.
We've got something dead center screen, drifting off at 9 o'clock. Okay. Again, this similar, it's like a white center with like really fine hairs coming out of it. Whatever they are, they're common.
Yeah, I think there's some kind of protist, single-celled organism for sure. There's, yeah, one up.
There we go. There we go.
See if we can get a good view of this one. It's floating above us.
Oh, neat.
It really is neat. You can see all these extensions, these pseudopod extensions that it uses to ensnare its prey. There it goes. Now it's all disorganized. That was nice. That was good.
Mm-hmm.
Little fish up there at the top. Another fish up at noon.
Cadignath, I think.
Yeah. I think there's more than one, well, many more than one kind of protist. Protists are extremely diverse, but we seem to see some larger organisms, larger protists of multiple kinds here. Mm-hmm. There's the one with the fine pseudopods in every direction, and then there's one that looks like it has 4 little extensions coming out that are a little bit thicker than the...
Maybe Cadignath at 6. What's this? Ooh.
That's good there.
A little jelly or something?
Oh yeah, it's definitely a
Ooh,
jelly.
neat.
Yep. There's a... like to see your tentacles.
I don't see any tentacles.
No, I'm not sure if it has them pulled in or just can't see with the light. I think, oh no, they're wrapped down. That's a It's a narcomedusa, like a Pagantha or Cunina type.
And that bright spot on it is probably an amphipod that's
nice.
taking a ride on the jelly.
Makes me dream of a white Christmas.
I want the screen closer to my face.
Yeah.
It's like I'm having a hard time seeing, like finding everything. Oh, that's something. Ooh, little fish.
Definitely.
Hi.
And now is that a lanternfish? I don't think that's a cyclothone.
Yeah, he has a broader head.
Yeah, broader head, bigger eye. I think it's family Myctophidae, which is another. This is a diverse group. If I have it right, but it's a myctophid.
Yeah, I think that's a really small myctophid.
Thanks, Mike. But this is great footage for a fishy person to.
Oh, neat.
Yeah.
So you think this is a baby lanternfish?
Yeah,
Is that what Myctophid is?
So. Called lanternfish.
Well,
because
I'm just guessing.
He has really long. Is that pectoral fins I'm seeing? Or is anal fin?
I don't see any development of the protophores on it, though, so I'm changing my mind.
Okay. Well, then.
There's a yellow spot.
An unknown fish at this point.
He has a really large mouth.
Yeah, it's certainly sufficient footage for someone to nail down.
Yeah, a fish person could tell you, maybe not the species, but what type of fish it was.
Mm-hmm. So we're pushing out again. There's some leeway to work.
Mm-hmm.
And we're about 10 minutes into this transect at 500 meters.
There's something flashing at 2 o'clock.
So,
Oh, it's off screen right about now. Yeah, it's probably a fish. Ooh, there's another one of those, maybe protists.
yeah.
There's one here with 4, it looks like. There it goes. Right off screen.
Yeah. I think those are phaedarians.
What is a phaedarian?
Single-celled protist, again, that was at one time classified with the radiolarians.
What is that?
Ooh, I think we have a medusa there. Come on, light. Oh, come back.
Don't the phaedarians sometimes form colonies?
I think
A few of them clumped together.
yes, that is my understanding as well.
Well, since it's so quiet, I'm going to revise my guess as to what that fish might have been.
Okay, Mike.
And embarrass myself. I think it may have been a juvenile owlfish, a bathylaggid. But again, we need to get a good frame grab to send to a fish person to find out for sure.
Bathylaggid.
They have big eyes like the, bathylaggedy. They have big eyes like like what I seem to remember from the brief video that we have.
Well, thanks for the guess. That'll give us something to something to assess when we send it out to someone. A little fish moving through the water column here. Okay. So that is not a jelly. That is definitely what I'm calling, what I think is a phaedarian. So this is this protist. Now we're going to find a nice big ctenophore in here somewhere.
Oh, no, that'd be cool. Oh, that's something pink. Another siphonophore.
Yep.
Ooh. Something at
that?
11?
Yeah. Oh, I think that could be a...
Shrimp, maybe?
That's what I was wondering, if it's a surgested shrimp, because they have these long... Yeah, it is.
Oh, cool.
Surgested. Beautiful.
And there's his antenna. That's long.
Yeah.
I'd imagine they're wide, so he could...
Yeah, that elbow in the antenna, that's very characteristic of them, and it functions sort of like the lateral line on a fish. It has sensory cells spread along that extensions to the antenna, so it detects water movement farther away from the body of the animal. So it sort of extends its detection ability out into the water around it.
And what was the name of that shrimp?
Surgestidae is the family. S-E-R-G-E. But you didn't happen to have a more specific identification on that surgested, did you, Mike?
No, I don't really.
Not a problem.
Send the video to somebody that knows them.
Sorry, I'll take a quick peek in the chat room.
I haven't seen very much chatting going on in the chat room.
Mm-hmm.
Nope.
Tammy Frank and Heather Bracken Grissom have been doing our crustacean identifications in the Gulf of Mexico. They have, at this point, looked at a lot of these animals.
It's great. I think they recently published a barcoding paper on crustaceans from the Gulf of Mexico. She and her group.
See
Yeah,
something.
Heather did a paper on
3 o'clock?
matching up larvae with adults using barcoding.
Oh, we got a jelly at 3 o'clock. Off screen. Never mind. Oh, it's gone. It went past.
What are those?
Yeah, the little thing at 1 o'clock, little flashing thing at 1 o'clock. Now it's center. Yep.
Yeah.
Now it's at 6.
Ooh, there goes a worm of some sort.
Ooh, little fish. He's tiny.
It's tiny or way out there, or both.
It's very silvery.
Gee. Okay, we are at the halfway point of our 500-meter transect. 20 more minutes to go. Yeah, let's take a look at that. That looks like a Medusa. Oh, just went off to the right. I think that was another Narcomedusa. Oh, there we go. There's this. It's a nice siphonophore. You can see it has its tentacles and then its front swimming bells, and it's rushing away from us. That was another one with two large sack-like swimming bells out front of it, not rocket-shaped. Nice.
I just saw an email from Stephanie Bush who says that the thing he thought was a pteropod that was either Cavalinia or something like that. She agrees that was either Cavalinia or Diacavalinia.
Oh, great.
The one with the beefy body.
The beefy body.
Yeah. I just sent her the call-in information, so maybe she'll be joining us.
Great. It'd be great to have Stephanie, another Steph aboard. Oh.
Yeah.
Hey, Alan,
Yeah.
you've been mentioning these eDNA samples that you're taking. What do we do with those, and where are those housed?
The eDNA samples themselves are going to go back to the Smithsonian and become part of the collections there. But as part of that, they're going to be processed, by which basically we mean that the DNA from the water sample is actually going to be extracted, and then we are going to amplify a marker, a genetic marker, that is something that's common to all eukaryote organisms. And then we will sequence that marker, and that will give us then as a profile of all the different genetic signature of many of the different organisms that are inside that we captured in the water column. But there we have a nice copepod coming into view. Most copepods are really small, and this is quite... Oh, wait, is that a copepod? No. Oh my gosh, I'm really not sure what we're looking at. I thought this was a copepod, but it's definitely not. Huh. Any feel for how the size of that? All right. It was tiny.
That was very tiny.
Well, that one had me confused. Mike, did you recognize that?
Well, it was definitely shrimp-like, and I don't know for sure which of the shrimp-like groups it was. There's shrimps, and then there's the krill, the thousands, and there's also a group that's one of the genera is Nathothalsia. I'm trying to think of the group's name. It's related to mysids.
Uh-huh.
It may have been a very small individual of that.
There's some marine snot. Yeah, when that thing got up close, I wasn't so sure it was an arthropod at all. Did someone call in?
Yeah, it's Stephanie Bush.
Hey, Stephanie, how are you?
And I missed what Mike was saying about that crustacean that you were thinking was a copepod until you got zoomed in. I didn't hear what Mike said because I turned the volume or the sound off while I called in. I thought I might get feedback and it would get weird. But yeah, like Glophogastrid, we used to call those things like mysids, but that might be wrong. But thing like that.
Yeah, that's the word I was looking for.
Glophogastrid. Cool.
Nathothalsia is a Glophogastrid.
Yeah, I would love to see one of those, actually. I don't know if have you guys seen one? Mike, do you recall if you've Okeanos has seen one of those off the East Coast?
I think over the years I've seen maybe a few, but not very many.
Yeah, I feel like they might be getting out of the way.
Yeah, we catch them regularly in the net. It may be that they avoid the RV.
Yeah, I would think so. But
yeah, we catch them regularly in the net. It may be that they avoid the RV. Yeah, we catch them regularly in the net.
There we have it. Whoa. Now let's take a look at that if we can.
How about
Happens. It's one thing disappears, another appears.
that? What the heck? All right, I'm going to make a guess. Siphon. No.
Oh.
It looks like it's squid ink.
That's squid ink. That's definitely squid ink.
Victory. Let's see.
It's close to being a squid, but not quite.
Yeah.
Signs that squid are here and running away.
I think I have a Medusa coming in at noon. All right, now it's about 1 o'clock. Yeah, it's out of screen there. Okay.
Oh, there's one.
Yep.
I just mentioned for the people that are listening that squid ink was in the form of what we call a pseudomorph. So they're capable of mixing their ink with mucus, and they can form a shape with the ink that looks about the same size and shape as the squid, so that if they're being chased by a predator, that the predator will attack the ink pseudomorph instead of the squid.
Mm-hmm.
And get a face full of mucus instead of a squid meal.
So tantalizing when you just barely see the shimmer of a Medusa and then it disappears. So many of the organisms out here are so transparent that they don't even show up very well in our lights. There's that thing that I've been thinking is a Phaedarian. But yeah, go ahead.
I've been thinking our lights might work better if they were at a different angle, but I assume that they've got them positioned the way they have in the past for these midwater guys. But a lot of times, lights on these transparent animals, if your light is going out forward from the sub, the transparent animals aren't going to show up as well as if you've got lights coming in from the sides.
So, did you say they'll show up better with lights from the side?
Mm-hmm.
Yeah, they will.
Yeah. That's how I like to photograph them when I get them in the... There was that Desmophyce again. Calicopher and Siphonophore with two big flappy swimming bells at the front of it.
There's something at 3 o'clock. It's bright. Yeah. Very reflective. One of those little fishes. Yeah. So we didn't collect any physical samples or biological samples in 1.
Yeah.
And I don't think we're going to collect in 2.
That's right.
I feel like we could have 3 buckets for the deep scattering layer.
Yeah, if we wanted to take a few during the deep scattering layer, that would leave a couple more. Once again, we have the capacity to catch 5, slurp up 5 different organisms on this dive today.
Mm-hmm.
And countless ones in the environmental DNA samples.
Here we go. There's 2 little. They look like X chromosomes to me.
They do look.
I don't know what they are, but they look like this.
Well, there we go.
Oh,
To
9 o'clock.
9 o'clock. I have a little
Oh.
Medusa there running away. Again, this looks like that maybe Tetrorchus. It's the 8 widespread tentacles. Oh, just sort of drifted off. Couldn't see the 4 gonads again, but that's the general shape of the animal that I would expect for Tetrorchus.
That seems to have a lot of tentacles for Tetrorchus, but maybe that's just different species just between what I'm used to in the Pacific and these ones. To me, and I'm not getting, as Mike said before, I'm not getting as clear of a picture here at home as you are, but it looked to me like a Colobonema type.
Maybe, but I think too many tentacles for, or too few tentacles for Colobonema, right? But you're right, Tetrorchus should only have 4 tentacles, and that guy had 8 tentacles. It's a good question.
We should have collected it.
Yeah, well, an allied Tracheomedusa is Ropolanema, for which the family's named. And that has 8 tentacles. And also, since we couldn't see how many radial canals were on that organism, that could fit. Thanks for mentioning that, Steph.
I was a little late. You mean on the collection comment? I was a little late.
Oh, no, you're fine.
Okay, because we totally missed it. It's way far away now.
Alan, as far as that last jelly, and there was one previously that you were talking about the bell being really reflective or refractive. That's what that partially made me think of Colobonema. But from my view, I can't see the bell at all. What I was thinking of is sort of the general structure of the tentacles, where the proximal part is very thin, and I can't really see it, whereas the distal part is quite thick. That reminds me of a Colobonema type.
Yeah.
Thing. But that is also true of Tetrorchus. Distal part is.
Right, but they're all in the same family, this Ropalinamatidae.
So it looks like the first transect was mostly siphonophores, and this one is mostly the bristle-mouthed fish.
Yeah, we're seeing definitely more of the fish.
There's something at 6 o'clock. Now it's closer to 9. It's right there.
Yeah.
Yep, it's got combs.
It's got combs, so that again looks like the Thalassocalaceae. Always a hard one for me. This is the sort of Medusa-shaped ctenophore. And this is the one that actually traps larger prey items. And that big Medusa-like cap closes over them, and then that's how it does its feeding. It's a very unusual mode of feeding for a ctenophore. Siphonophore floating by.
Two minutes left.
we've got Heather Judkins in the chat now. She's giving an invertebrate zoology test.
Oh.
All right, come on.
What was that?
What was that?
Neat.
Hopefully we'll see it again. It's another one of those protists. More fish swimming by.
There's something at 9 o'clock. Another one of those hairy protists.
Yeah.
Looks like a little fuzzball. The in a transect? Is that what I heard?
So our transect is done at 500 meters. The next transect is going to be in the deep scattering layer.
Yeah, so right now we're still at 500 meters. What's going to happen is the ROV is going to set up to make a move, and then we're going to drop down to 600 meters and be in the deep scattering layer, and then do our third transect. Hopefully maybe collect if there's anything there to grab.
Can you check the EK signal to see if the deep scattering layer has descended at all since you targeted 600 meters?
Yeah.
Stream
I think
3
I'm-
is full screen, EK60.
It looks like it might be deeper.
I would expect that it is deeper than it was, a couple hours ago.
Mm-hmm. I'm seeing a red line. Hang on. Roland? I'm seeing the red line on the EK. Is that us? Or is that the deep scattering layer I'm seeing?
Hey, Steph. I hear that's maybe like a false return, but it does
A
look
false
like
return?
the deep scattering layer is in that region.
Okay, so it's the light blue that I'm seeing behind that red line.
Yeah,
Okay.
and then it looks like it extends down to the 750 meter
750.
region as well.
Yeah.
There may be like some false gaps there too. It's kind of imperfect.
Oh, okay, I see that. So that false return is actually that
So
red.
the deep
Okay.
The deep scattering layer is actually not a single layer anyway. It's multiple layers of different species that have their own preferred light levels. So it could be more than one.
Mike, if you turn on
in the evening when they start up,
if you turn on feed 3, you can actually see it.
Up, they do. I'm sorry?
If you turn on feed 3, we actually have the EK being sent out. So it looks to me like it's still in that area.
Okay.
It's just spread out a lot. So instead of it being more focused in the 600 to 25, it goes all the way down to like 750. So it just seems to spread itself out a little bit. And there's a false return at about 6. You see that bright red false return?
Yeah, I do see the bright red.
So it's in between. You can see the light blue that kind of sits behind it. It's looking like there's almost two, like it's separated. Yeah, if you want.
Well, it's not unusual to see multiple layers sort of concentrated into a deep scattering layer.
Mm-hmm.
I guess we just picked whichever one is the strongest signal.
We're going to do 700 too. Yeah, I think so.
Yeah.
That works. Yeah, it looks like we were going to do a 700 one anyway, so if it just happens to be in the bottom half of that deep scattering layer, then yay, that works. So I think we're going to stay at 6 and do another one at 7.
Correct. And we will collect eDNA at the beginning of the 600-meter transect, but not collect the eDNA sample for 700 so that we can save the remaining for our 900 and 10 meters off the bottom.
So just for anyone watching on the quad view, my cursor is about the 600 depth right now, which would be on the upper portion of that deep scattering layer that we're seeing. And then 700, our next transect will be down here. So both of those should be within that scattering that we're seeing on the 38.
Thanks, Matt. We're at 590 meters.
Getting close to our third transect depth as we're here on the Blake Plateau exploring the water column.
Yay.
The largest biome on planet Earth, encompassing
String 3 back to
about
multiview.
95% or more of all the livable space on the planet.
When are they going to put condos in?
There's all sorts of...
Would that be the larvation houses?
Yeah, the larvacean houses, just what I was thinking.
You beat me to the joke.
There's something.
It's flapping.
Yeah, it's
like a
crazy.
pteropod, maybe? I'm afraid we didn't.
No, I don't think we introduced ourselves.
So-
So if you're just tuning in and you haven't been out with us this cruise, I'm Stephanie Farrington. I work at Harbor Branch Oceanographic. I study deep-sea coral reefs around Florida and in the Gulf of Mexico and all the way up to South Carolina and around Cuba. And I'm with Alan.
Yeah, Alan Collins, here of NOAA Fisheries and the Smithsonian National Museum of Natural History, where I'm curator of sponges, some of the cnidarians and the comb jellies. And
This is Windows to the Deep 2021,
And
NOAA Ocean Exploration.
And we are out over the Blake Plateau, actually at the far eastern edge of the Blake Plateau, over 1,080 meters to the bottom. And we are doing midwater transects at 300, 500. We're about to start our transect in the deep scattering layer at 600 meters. And from there, we will continue to proceed down in the water column for transects at 700 meters and at 900 meters. And the final one of the day will be 10 meters above the seafloor.
Yep, we're just waiting here to get a Niskin bottle to get eDNA sample right before we start transect.
Who do we have on the line on shore?
On the line, I have Mike Vecchione. Mike, can you introduce yourself?
Well, you just did.
Oh,
I'm Mike Vecchione. I'm the squid guy.
The squid guy, yeah.
I get paid by NOAA to work at the Smithsonian Natural History Museum.
Nice.
Best job in the world.
Yeah, right. Except for mine, where I get to be out on a boat all the time.
Then we also have Stephanie Bush, I
Yeah,
believe,
and then-
online.
Yep, this is Stephanie Bush. I am also at the Smithsonian National Museum of Natural History, but I am not NOAA. I work at the Smithsonian. I just straight work at the Smithsonian. In the same department, though, invertebrate zoology, as Mike and Alan.
And Stephanie has been out here to the NOAA ship Okeanos Explorer.
All right, so that was sample 3. We just popped at 1512. And I think we're starting Transect 3.
You are correct, Alan. I was on the Windows to the Deep 2018. So in visiting similar locations to where you guys are now, near on or near the Blake Plateau.
All right, start Transect 3. So this is our 600 meter deep scattering layer.
Yes. Now let's see what we see. And this will be another roughly 40-minute, or precisely 40-minute transect.
I see something. All right, we've got some kind of jelly thing dead center right above center screen. You can zoom right there.
Good eye, Stephanie.
He's moving like a squid, but I don't. No, siphonophore, yeah?
Yeah, it's definitely a little siphonophore. Trying to see.
Oh, neat.
Don't see the...
Looks more like a doliolid face.
Oh, good for you. It is a doliolid with little, it's got it's reproducing itself.
What is a doliolid?
That is a pelagic tunicate,
Oh, nice.
and it's a relative of the salps. And it sort of superficially looks like a siphonophore, but it is not. It's a filter feeder. Now, we can start thinking about possibly collecting if we find organisms of interest down here.
There's something.
Schmutz.
Yeah, loose tentacle or something. Got another one of those bristlemouth fish, maybe? 1 o'clock? Oh.
We've got George Matsumoto now from Mbari on the chat room. That'll be nice and helpful when we find things to look at.
I see cadignaths and those little white fuzzy protists.
Yeah,
You see a few of those?
Phaeodarians, radiolarian type things.
And the bristlemouths is so far
Lots
really
of
what we've been seeing.
cadignaths. And the one doliolid with its reproductive zooids dangling off the back.
There's something center screen. Yeah. There he is.
There's a nice cadignath, arrowworm, common name. Just tough to get the focus.
There's something right.
Another.
Siphonophore maybe? Oh!
Oh, just went by on the upper left. That looked like another.
Protozoan.
Yeah.
Yeah. There he is. Sweet.
Yeah.
Look like
Foram or radiolarian.
little pom-pom. Looks like something I'd hang off my Christmas tree. Oh, something dead center, out of focus, and it's gone. There's another one. Yeah, it looks like something out in the distance a little bit. It's very disorienting.
It is.
Here's a little white thing. Oh, fish. Is that another bristlemouth?
I'm not sure that was a bristlemouth.
He was swimming differently.
Yeah.
There's been a request to comment on the light pool on D2 compared to our typical benthic setup. Normally we would have swing arms all deployed and trying to light up as much of the seafloor as possible. Right now we have all of our light focused into a sphere of light within the viewable range of the Zeus Plus, so that way that column of water is all lit up. We usually try to avoid doing that because backscatter is usually a bad thing when you're trying to look at an animal on the bottom. But that's what we're shooting for now, is lighting up more of that water between the camera lens and out as far as we can, so that way when an item comes into the view we can keep it lit for as long as possible so that we can see it, get it in the right spot, and then get a zoom on it.
What is this?
We don't have a way to light from the sides very well, but we do light from above rather than straight out from the vehicle.
That's definitely a comb jelly, Thalassocalus, I think. Again, the Medusa-shaped one, which we've seen several times today.
Any interest in collecting it? Think it'll hold up?
It would be good collection at some point, for sure. It's going to be very difficult for us, but we could try our Rain-X preservation. They're very delicate animals.
Do you think it sucking into the tube would probably destroy it? We have to a clamshell instead.
It might.
Yeah.
But I don't know for sure because I haven't tried. Yeah, George Matsumoto confirms that was Thalassocalus. Not sure of the exact proper pronunciation, but that is the sort of modified lobate ctenophore that has it's more like has a Medusa shape to it, where the lobes have grown together, and that's sort of has like more of a bell-like structure. It's also unusual compared to other ctenophores in that it's not encountering the environment with its mouth first, which is typical of most ctenophores. Mm-hmm. It's another one of these beautiful protus, little pom-poms. Mm-hmm. George Matsumoto likes my pronunciation of Thalassocalus.
What should it be? Is it wrong? I mean, it's not. It's
I think
all.
That's the way he says it as well.
Okay.
I often get that question from students. It's like, how do you pronounce this correctly? And I tell them,
it's Latin.
It really varies a lot from
Yeah.
person to person.
It's Latin. There's no, like, it's a dead language. There's no,
Yeah. There's sometimes the actual proper Latin pronunciation, like for Cnidaria, that's Cnidaria, right? You know, so that's like, it would be something that doesn't sound right to our ears, but could be correct. All right, so I'm thinking that's a siphonophore.
It's out of focus.
Drifting to the left.
Got something at noon.
George suggested that siphonophore looked like Nanomia. I think we've seen some similar looking siphonophores starting at the 300 meter transect, and that sounds reasonable.
Yeah, let's see. I think that's probably just sea snot.
It's good.
Yep. Somebody blew their nose to the water column.
That's somebody else's food as it drifts through the water column toward the bottom.
Here we've got something at 3.
Yeah, is that a little jelly? Oh no!
Ooh.
So we have our first. Yeah. This little Mesocordius. Could we collect this by any chance? Oh,
Ooh.
maybe it's going. At some point, we're going to want to collect one of these larvaceans, one of the larger deepwater larvaceans, either Mesocordius or Bathycordius. That's one of our targets.
And what did you call that last one?
It was a larvacean. And basically what we could see was a mucus house that it builds. And towards us was the tail of the larvacean, and it was beating to create a current, and that pushes water through the house, which is a really complex structure. It's got inner part and outer part, and it filters so that it just gets the size particles that it wants to eat. Nearby, its mouth.
It's a swimming mouth.
And then it, basically, it's got a big long tail and then and a head region, and then it will discard the house and make a new one.
Oh, neat.
Someone turn me off. Okay, there we go. This is a nice Trachymedusa, a Rhopala nematid. I think it looks like Rhopala nema, maybe. It is beautiful. You can see it has its 8 tentacles deployed. It is one of these jellies that will sit and have its tentacles out looking for food, and then it will also have an escape response if we were to come closer to it, where it will beat vigorously and swim away. Really, really nice video of this animal. So I think this is probably the Medusa that we were seeing before, but you can see that this one has the really prominent gonads, sort of 8 of them. These are along a radial canals, and in the central area there's a throat-like mouth dangling down in the middle. Thanks very much for that great video.
Yeah, great shot video. That was good zoom.
Yeah, and George confirms that that's Rhopala nema. So we're going to push forward again.
For anyone just tuning in, NOAA Ocean Exploration is on board the Okeanos Explorer, and we are about 600 meters down in the water column off of Georgia, and we are exploring the deep scattering layer. That's the zone where there's a lot of little critters floating around, and looks like we have some stuff right here.
I think that's snot. So
Snot.
marine snow.
That is
Accumulation of mucus.
often considered a good carbon pump into the seafloor.
Exactly.
Also could be food for something else at another point. Maybe that copepod, something above it. And we're going to keep making our way down in the water column on these transects, step by step. There's a nice...
All right, so we have a lobate ctenophore. Oh. Wow. Oh my goodness. That's incredible. Definitely get some good footage of this.
It's like you can see the cilia beating on it, like little light refractions.
Yeah. All the ctenophores have these comb rows, these giant cilia, and when they beat, they refract the light, which we can see here.
Yeah, it's not bioluminescence. It's actually a reflection.
Yeah.
Refraction.
This might be a nice collection. George Matsumoto also, since we have a really good video of it, I think I would, if it's possible to collect that, I would like to do that. Yeah. Yeah.
Just change stream 3 for sample mode. You can see 4 different cameras. You can see that on stream 3.
Oh, it's big. And it's in. Yes. So it's 4th sample. Confirmed. Is it canister 5? Great. Thank you so much. That's really exciting. So just this past year, a paper, ctenophores are notorious for being almost impossible to preserve, and many of the deep water forms especially are incredibly delicate. And but a paper was published this year suggesting, showing actually that Rain-X of all things can be used as a preservative for ctenophores, and so we actually brought some Rain-X aboard, and assuming that that animal has survived that its body has survived going into the canister, we will try to preserve it here on board as well as a subsample for to look at its genetics.
So the same stuff that makes water fly off my windshield also makes water fly off of ctenophores?
Yes, ctenophores and other soft-bodied organisms. It was used and shown to be quite a very effective preservative for morphology. So that was great. Really excited about that. Yeah, so that ctenophore didn't really match up with anything that I'm familiar with, and George Matsumoto, who also has a lot of experience with ctenophores, likewise is not exactly sure what that one was. So let's take a look at that. It's another one of those protus pseudopods extended. Thanks. So these lobate ctenophores, like the one we collected, they typically have their lobes out in front of their mouth area, and they have tentacles along their mouths. Not all of them, but most. And prey gets sort of pushed in till it gets stuck on the inside of the lobes, and then eventually moved to their mouths. it's a really different form of feeding than the other the other typical style for ctenophores is that the ones that deploy these long tentacles, often with side tentacles, more like jellies and siphonophores. They're, casting a net and then waiting for organisms to bump into that. Take a look. Something in the water. Okay, I don't know what that is. Oh. Oh, I think that might be, well, I'm not sure, but look like a radiolarian, yeah. You see them as sometimes looking like that in balls, but I've never seen one with a sort of a fishhook shape like that.
Yeah, it kind of looks like a sad pyrosome.
A sad pyrosome. Yeah. George confirms that was a radiolarian. A rad. Oh, I think a little siphonophore went by. So we are in the middle of our third transect of the day, third of 6. About 10 minutes left to go in this transect that's taking place at 600 meters in the water column off on the eastern edge of the Blake Plateau. It's interesting that Mike Vecchione in the chat room is commenting on the sort of, if we're in the deep scattering layer, this is not very not as densely populated with organisms as we would generally expect. And I have to agree. But we did get that that the deep scattering layer was sort of spread out. Not in a narrow layer, but going down, I think, to nearly 750 meters. So we'll still be doing the next transect at 700 meters, which will be in the same general area. And who knows what that'll bring. Take a little zoom in there. Okay. There's something in the water just floating by above us.
These animals are incredibly adapted not to be seen.
They're doing a good job of it. That's more another radial area or 4M in the water. Another pair. Another nice image of a 4M. They seem to be the most abundant thing in this transect.
Can we zoom on the test, the spot right in the middle?
Nice.
That is beautiful. I remember studying
That is
the
nice.
shells.
We should mention for the people listening that little test is a hard piece of calcium carbonate, and they form much of the sediment of the deep sea. When they die, they sink down and accumulate on the bottom as what's called foraminiferal ooze. So not only is that contributing to the sediment, but it's also moving carbon dioxide, again, out of the near surface waters and down into deep water. But they're really important ecologically.
Yeah, Mike, I remember in my marine geology class sorting through 4M shells with a paintbrush and looking at them under a microscope to determine the age of cores, geology cores in the sediment. It's a great carbon pump and a way to date sediment layers.
They're also used to determine the overlying water masses of an area geologically. So, when you look at really old sediments, they infer what the temperature of the water was, both from the species of 4Ms that were there and from the oxygen isotopes in the shells.
Sounds like paleoclimatology.
For determining, climatology.
All right. Yeah, this is what I've been calling a Phaedarian, but now it'd be nice to get a good close-up of this to see. Since we've seen quite a lot of these. That was a really nice video, though. Should allow us to be positive about what they are.
Hi everyone, we just finished our 600 meter transect. So the ROVs are going to stop now and reconfigure and move down to 700 meters, and they will start the next transect in a few minutes.
It's a good time in between transects to just do a quick recap. This is NOAA Ocean Explorer, and we are on the Windows of the Deep 2021. We are at 600 meters of water off the east coast of Florida along the Blake Plateau. We are actually floating above about 1,080 meters above the bottom doing midwater transects today. We are 171 miles from shore, so pretty far away. We did a 300 meter transect, a 500 meter transect, a 600 meter transect, all of which had water samples before the transect started. We're going to be going to 700 meters, which seemingly looks like it's still inside the deep scattering layer. Hopefully. Yeah, I think so. Yeah, roughly. Bottom of the deep scattering layer. And then we'll do one at, I believe, at 900 meters, and then another one at 10 meters above bottom. So we've collected one sample, which was a ctenophore.
Beautiful ctenophore, yeah.
Yeah, plus 3 waters so far, and we're going to do another water sample at 900. We're not going to do one at 700 because we don't have enough bottles.
And one at 10 meters off the bottom.
Yeah.
Yeah.
So just a nice recap real quick. We are descending. We're at 620 meters. When we get to 700, we'll set up for Transect 4. We're crossing 650 meters.
I'll put the K60 full screen on stream 3 as we transition.
Approaching 700 meters. There we go. We've settled out at 700 meters. All right, so we're just, go ahead.
So we have arrived at 700 meters, and now the pilots are going to swap out and get some fresh bodies in there. We're right in the middle of lunchtime, so we've got to make sure everyone gets some chow. And then we're going to proceed on our next transect at 700 meters, which is on the lower end of the deep scattering layer.
Mm-hmm. It looks like that deep scattering layer is concentrated a little bit shallower than it was when we started this morning. So it looks, yeah, like when we were coming down, it looked like the scattering layer had separated a bit.
It doesn't look like it was avoidance. Yeah, it looks like it just happened because the ROV was higher before, and the scattering layer actually went towards the ROV.
Yeah. I think it's probably light related as opposed to like our influencing its location.
Yeah.
We're just prepping to set up for transect number 4. As we shuffle pilots here for lunchtime. I don't think so. Pilot, this is science. Did you guys start the transect yet? Okay. That is. Ooh, there's something. That's neat.
That's nice.
Hello.
Oh, wow.
My guess is siphonophore.
We'll go with that guess. Yeah. All right, now,
Oh.
no. Washing away.
Okay, watch lead and shoreside. This is the start of the 700-meter transect. We got 1607 UTC.
Thank you. So we're starting transect 4 at 700 meters. Bristlemouth. Even though that looks a lot like the other one with that gold side.
Hey, everybody.
Another one of those protozoans.
Yeah, that's foraminifera.
Ram.
We got some great footage when you stepped out.
For lunch.
Yeah.
And you caught something.
Yes, we caught a lobate ctenophore.
Oh, neat. Wow, what is that?
What do we got there?
Looks
Is
like
that
one
a
of my
fish?
knitting needles. A sewing needle. What is that?
I think it's a fish.
Like a pogo fish.
Yeah, I think if you zoom on the bottom part of it, you'll find that it's got a head of a fish.
Mm-hmm.
Like a barracuda?
There's a couple of different things that are long and skinny like that and stay vertical in the water column. So you have to look at the tail and at the head.
There's another siphonophore.
Nice.
Let's get a good zoom on him.
Yeah, I think that's that same one, sort of like a Praed. It's got the two large swimming bells out front of it. I think we could tell the genus apart if we get a close look on the front swimming part at some point. Thanks. Here we have another Ketignath arrowworm.
That's a good shot.
Really beautiful shot of it. These are hermaphroditic animals that sit and wait predators, mainly feed on copepods, and they have a potent neurotoxin that they deploy when they bite their food.
So they are venomous.
They are venomous.
Looks like another one ahead of us.
I
There
think
we go.
that's
Another
we've got another Ketignath, exactly.
There we go. So I would assume this is bright dot is his eye. Is that the case or no?
No, I don't think so. I think its head is at the other end.
Okay.
Yeah. Those must be
The
its testes or
photophores?
gonads of some sort.
Okay.
I think that's the gonad at the tail end.
Oh, now I
There's a
see.
pair of them, actually.
Yeah, like his head is more of like a fuzzy white.
Yeah.
I don't think they have eyes.
I don't think they're blind. Just they don't even have a light sensing organ.
Well, I don't remember all of them. They don't have any discrete, large eyes.
Okay.
They might have photosensitive organs of some sort.
Mm-hmm.
Not sure how they find their prey.
Yeah, I think they sense the movement in the water. But not sure how they do that either.
Ooh. I think that's probably just
Schmutz.
sea snot. Yeah. There might be something here at 9. There we go. Oh, that's neat.
Yeah.
Siphonophore?
No, I think that's another doliolid, or it's definitely a pelagic tunicate.
Awesome.
Yeah, this is really nice. Good footage.
He's similar to body form of like a sea pen,
Yeah.
right? With like the bulb at the bottom, and then
With all the different zooids around.
totally wrong phylum, but that's super neat.
Ooh, let's watch it swim a little bit.
Ooh.
There it goes. Oh, yeah.
Awesome.
That is really cool. So the front end there it's the mother. These are a bunch of little babies that are growing off the back end.
Will those butt off and become
Yes.
independent?
And the front end has these circular muscles, which is
Makes it so it makes that pumping action and can move itself forward. Obviously limited, but
gosh, I've never seen this.
you want to collect it?
No, I think that's all right.
Okay. Okay.
Understood. That was really, really nice footage of that animal. Thank you.
Everything looks like a siphonophore to me out here.
Huh.
I think that's going to be my default critter.
That's okay.
When in doubt. Better?
The amazing is out there. It's like, there we go.
Oh, neat.
Oh, there we've got a nice sidipid ctenophore. This would be a collect if you're... It has really fine tentilla off the side. So this has the two long deployed tentacles, and then off the side of that are very small, fine tentilla.
We're going to try and get him?
Yeah. If it's doable, that'd be great. If it's not, that's okay. We can just get as good footage as possible and leave it.
So that's going to be sample number 5.
That would be sample number 5 if we can do it. I'll look at it turning. Oh, you guys. Oh, it's trying to run away.
It's not very good at that.
Yes. Yes. That is great.
Ooh.
Canister 4.
Yep. So that's sample 5 went into bucket 4.
Did it come apart? Mm-hmm. Oh, that's great. Thank you very much, pilot. I think it was. Can't believe we just asked for 2 really fragile guys. That's okay. We're going to have to put that in as Sidipida, but maybe we can do better when we get a look at it. That's one of those nonsense taxa that doesn't actually, we haven't sorted it out yet.
Yeah.
It's a group of many different, not necessarily closely related organisms that have sort of a similar body plan.
And they're probably pretty few. I would assume there's not very many samples of them just because they're hard to get to.
Yeah.
And they're not coming up in, copepod? They're not coming up in nets. You know, or if they do, they break up.
There's a beautiful copepod.
Little fat guy.
It is.
Keep swimming, bud. Ooh, little ugly guy. He's so cool.
Oh, he's awesome. And most of the copepods that have been in the water column here, they've been so small we haven't been able to get a good view of them, but this one is nice.
Yeah, I feel like you're wasting a lot of energy, man.
Lots of copepods in the midwater are vertical migrators, part of the diel vertical migration that happens every day all over the planet. Going up to the shallow waters at night, we're in the absence of light, and make it difficult for the visual predators to get them, and then returning to the deep, dark waters during the daytime.
Yeah.
Oh, yeah.
That's
The answer to
different.
that question is always yes. So now that's...
Oof. I think. Oh.
That's similar to what we were looking at before, but with a lot more tentacles. But I think it's also maybe that same Rhopalendomata day. Oh, it's nice. So we have 2 ctenophores among our samples. We may want to diversify, but
Yeah. There's nothing. I'm afraid to collect this stuff out here just because it's so fine.
I know.
It just can break so easily.
We're going to give ourselves some good work later today.
Another copepod, maybe? There. Oh. Might have been a fish. I don't know. That one's a fish. Yeah.
Is that cyclothony?
Nice. He has that red or pink like patch. Is that bioluminescent, or is it just? Seems like it should have a purpose.
I don't know.
Looks like there's something center screen. Yep. A ctenophore.
Yeah, I think that's probably the same. It'd be nice if we could have some video footage of this. Look how fine those and delicate those side tentillae are on this. Really, really, it's almost hard to imagine that they can pull anything in. It's just like almost like a cloud.
Yeah.
So that would be consistent with them eating really small organisms out of the water column. Wow. And those tentacle sheaths are right on the outside of the animal. So the mouth side is up.
Oh, facing up?
Yeah, it's facing up. Yeah, it's typically the way ctenophores. And this guy is deployed. Basically, it's sitting and waiting. Now it's capturing food, and it will, when it's got food in its netting, in its tentacle nets, then it will swim away and bring the tentacles in and put them probably adjacent to the mouth so that the animal can eat that food that it's caught. Wow, it's just really something else.
So this, I would assume, would sting you if you touched it.
No, so comb jellies don't have stinging cells like cnidarians. Mm-hmm. They have sticky cells. So they have these cells that can secrete a very strong adhesive, and that's how they actually wind up capturing organisms.
It's like a fine spider web.
That one was really fine. So some cydippid ctenophores have really large tentilla. Mm-hmm. So they have the main tentacle and then tentillae that can be really long and strong for catching large crustacean mm-hmm. prey. Those are just so fine.
Perfect for midwater.
Oh, yeah, interesting. So George Matsumoto suggests they could be larval lobates. So the lobate ctenophores that don't have the tentacles, mm-hmm. they have larval forms that are this cydippid, basically body form with the two tentacles.
So
I didn't know that they could be so large as that.
Something at 4. All right. There you go. Another ctenophore. There you go.
Yeah, we have a lobate ctenophore there.
Now this one doesn't have those tentacles.
No, this is the kind that has the lobes up front. It's moving through the water. It was mouth down and moving through the water, mm-hmm. mouth first.
We got something ahead, 2 o'clock. Yeah, one of those like little needles. That's a fish, likely.
Yeah, I think that looks like the fish we were looking at before. Is that a snipe eel, maybe? Or?
No, it's not a snipe eel.
What is that, Mike?
I should be able to. I have to do some digging before I embarrass myself.
Ah.
Is it Avicentina, or am I thinking of the wrong thing?
Can we get a zoom in on the bottom, the very bottom part of it?
Can you guys zoom in on the head, the bottom? They're at max zoom, Mike.
about. Okay.
Yeah.
I'm thinking something like Sarivomer, but I don't know for sure.
Cerebomber was my other guess, but I know that I confused the two. But I think Cerebomber.
Thanks, Steph.
I'm going to need you to go through all these C-tube notes with me because I have not spelled a single thing right.
I will definitely go through them.
Got something center screen. It's drifting a little to the 9. There you go. Oh, it's just schmutz, I think. Yeah. Sea snot.
Old Larvasian house, I think.
Maybe something coming up at 10 o'clock, 11 o'clock, 10. Well, I'm sorry, 8 o'clock. My clock is wrong today. Oh. No.
Small. Is that a copepod? Nice.
Oh, yeah.
We're a little over halfway through the 700-meter transect.
Awesome. There's siphonophore drifting off.
Yeah, bottom right.
Yep. There's like 4 amps.
Another 4M, yeah. Is that a Keatingnath?
I think so.
Yep,
There
another
you go.
arrowworm. See the gonads in the back end, the top, and its mouth is just at the bottom there. Come out, wherever you are. Just a sinker.
Another 4-amp, looks like.
Mm-hmm. Been really abundant today.
Yeah, it seems like they're in almost every depth, too.
Mm-hmm.
They're not really regulated by, or segmented by depth zone.
Pushing ahead. Fish, bottom right.
Ooh, that's a new one.
Oh, yeah.
He's really dark. Any idea what that was? Fish. I
Yeah,
had his
fish.
Spine, so.
Yeah.
The same way.
Look like
A dragonfish. of some sort?
dragonfish, yeah.
Okay.
Yeah, I agree. It's one of the black dragonfish.
So a stomiid, if you're going to put it in the chat or the annotations.
I'm going to have to go back and correct every
Can you say
single
that one more time?
Every single one of these. Estomiid?
Stomiid, so they're the family Stomiidae, I believe, is includes the dragonfishes. One of the genera is Stomius, but I didn't get a good enough look to try and identify down to genus.
Yeah, there
Oh,
it is.
there's the siphonophore. That's a calicopheran.
Stop moving. The Cadignath, maybe? A little fishy. Oh, he looks different.
See if we can, maybe we can get some video of this guy. Oh, well. He was there, he was gone.
There's another siphonophore.
Yeah. Oh. Yeah, go the swimming bells.
That looks almost skeletal, even though it's definitely not.
Almost.
Oy. He's still there. He's just out of focus.
That's definitely another calicopheran.
Ooh, that's neat.
That's a Larvacean house. We can see if we can see the Larvacean in it. Do you see any movement, beating of the tail? There it goes. I don't see the animal there with it.
Abandoned home. Whoop. Bye.
Yep, but fairly freshly abandoned. It was still pretty organized.
Mm-hmm.
There's one kind of ctenophore that specializes on eating those guys. Another kind of lobate closes around it and then waits for the little animal inside to try to escape, and that's how it eats it. It's all the eat or be eaten.
Yep.
More likely eat and be eaten at some point.
Eaten and then eventually be eaten.
Yeah.
Yeah.
Schmutz with a
Schmutz
fish
and a
going
fish.
by.
So reflective. There's somebody. Oh, neat.
All right, so what are we seeing there? No comb rose, not a ctenophore. The other thing that I don't know.
A partly consumed jelly, like Medusae?
Maybe.
Weird.
Or I always think maybe a siphonophore bit when I see something really odd shaped like that, but it's so...
Now it's looking more salpy, although I don't see any muscle bands. So, yeah, I guess nectophores, the siphonophore, maybe.
I have no idea. I'm like at an absolute loss for words here.
That's
I don't know what it is.
good video of it.
It's very odd.
Does that help?
Ooh.
My guess is south or like a solo doliolid without the nurse chain behind it.
Interesting.
2 more minutes at the 700-meter transect.
Thank you.
Yeah, George Matsumoto also agreed that that was a doliolid. I couldn't see the muscle bands, but interesting and really great footage of it. Let's see if
Kid Ignatz again. That's new. What's that? Tina 4?
yeah, I think
Look
that's
how neat.
definitely a Tina 4 with little short comb rows. Oh, I've seen that somewhere before. Oh my gosh, I'm gonna ask for another Tina 4 collection. No, let's get a good video of that. So.
You've got the Rain-X. I think you should ask for as many Tina 4s as you want.
That's true. Diopia, maybe.
Whoop.
Wow,
Awesome.
that was really cool.
We'll call that the end
End
of the
transect.
700-meter transect.
Great.
Thank you.
And we'll be taking an eDNA sample at the beginning of that.
Ooh, what's that? Ah! Tina 4, did you see it?
Oh gosh, no, sorry.
You can watch the replay
Yeah,
in the clipping station.
I will watch the replay. Oh yes.
That's different. That looked like a shrimp to me. There he is. Little shrimp.
Oh, he's gorgeous. And he's gone.
And he's back.
That was nice.
Bye.
Yay, things are looking up here.
I think that's a suggestive.
What kind?
Suggested shrimp?
All right, we're at 725 meters. And we're dropping down to 900 to get Transect 5. And then we have what? Transect 6. And then we should be done, right? So that's 10, 1,000. We have a 900 meter.
Yep.
And then a 1,070 meter.
And we'll be heading for our 4th eDNA sample collection just prior to the start of our 900 meter transect.
No, it won't happen until we're at 900. That's okay.
So if you're just joining us, this is NOAA Ocean Exploration Expedition EX 2107, Windows to the Deep 2021. And we are on the eastern edge of the Blake Plateau, investigating the organisms of the water column through transects that we've been taking at 300, 500, inside the deep scattering layer at 600, and then we've just recently concluded our 700 meter transect, and we will be going down to 900 meters for a transect, and then down to 10 meters above the bottom.
Yeah, please, above the bottom. We don't want to go
not below the bottom. The bottom is at 1,080 meters. So that's actually our little peek into the bathypelagic.
And then we might go to the bottom and just take a peek.
Yeah, if we have
time left.
If we have extra time before the end of the dive, we'll look along the bottom and see what we can find at that depth. Here, very close to the Blake Escarpment, which is the edge of the Blake Plateau, where it drops off quite quickly and quite deeply. This expedition is to investigate and explore parts of the Blake Plateau that haven't been looked at before, and then also several spots along the Blake Escarpment, including the Blake Spur, which is a big, feature that sticks out farther to the east, and we'll be investigating that wall as well.
Yeah, there's very few dives out here. It's really mostly unexplored. We do have some stuff closer to the land, but way out here where we're, 175 miles from shore, there's hardly very little data. So this is mostly exploration.
Yeah. And so you've done a lot of work with the coral mounds and bioherms.
Yeah, the past few years I've been doing lots of mesophotic stuff, so much shallower. But yeah, we've done some work on the Lophelia reefs, and John Reed, my boss, has done a lot more work on the Lophelia reefs than I have. But once our submersible, the Johnson Sea Link, was decommissioned, we really haven't been able to get out too deep. So I've really been restricted to mesophotic stuff lately. Yeah, past couple years. Working the Gulf, and we did get to circumnavigate Cuba, which was awesome.
Very nice.
Yeah. That was awesome.
So most of our dives are planned to explore the benthic habitat, but this is one of 2 full days dedicated to investigating the animals that live in the water column. It's of course the largest biome on the planet. 95% of all livable space is in the midwater, and these organisms are really important for their connections and how they make the rest of the marine ecosystem go. So a lot of the zooplankton that live here are food for large megafauna, whales and sharks and tuna and sunfish, things that we care about. And also, they themselves then are, of course, in their own complicated food webs. And they transfer nutrients and carbon and plastics, all sorts of things. They help move those to the deeper ocean.
We don't want plastic moving to the deeper ocean.
No,
But
of course we don't.
that.
But it is doing that.
It is doing that.
More and more papers are coming out showing that different organisms are ingesting microplastics. A couple recent paper on showing several jellyfish and comb jellies that are ingesting microplastics, and then
It gets into the food web.
Yes,
Can actually
exactly.
affect people, I would imagine, into our food supply. And plastic doesn't biodegrade. Plastic only photodegrades. So the only way it breaks down is from the sunlight. And it slowly breaks down into smaller and smaller and smaller pieces until it becomes a microplastic and then gets ingested into the plankton. And I believe in the Pacific garbage patch, plastic outweighs plankton by 6 times.
I did not know that.
Yeah, it's pretty gross. So recycle, everybody. We're about at 900 meters now.
Yes, we're approaching the depth of our next transect and our next environmental DNA collection.
All right, we're at 900 meters.
Oh,
Look
there
at that
goes
guy.
a little narcomedusa to the right.
And
And 4 tentacles, sort of Aegina-ish.
he's off in the distance now.
Mm-hmm. So we're getting into position.
Well, it takes a
And
few minutes.
we'll be making the eDNA collection right at the beginning, just at the beginning of the transect. And the eDNA collection is essentially just a water collection, and that water then also has loose skin cells and gametes and even loose DNA that the organisms in the surrounding water have shed. And so it's we can basically extract that DNA from the water and then sequence it and get from those sequences an idea of the organisms that are in the surrounding water. So
It's like population genetics. Ooh, what was that? I think that was a worm.
it did look like a worm, like a
Polychaete. Here we go. Here we go.
there's a nice looking jelly. I think this is Solmissus.
Neat.
It is beautiful, absolutely beautiful. So this is a narcomedusa, which are pretty distinctive because they have their tentacles coming out of the top of the bell. Appears to be swimming away from us, and you can see it's what's called the velum. That's what's pulsing here. So
It's like clear skirt.
it's a skirt, exactly. And a lot of hydromedusae, which this is, that velum is stretched more taut and is used more like a jet for jet propulsion. But in Solmissus and some of the other Narcomedusae, it's almost facing straight down towards the camera rather than being sort of sucked in towards the middle.
Does he have a copepod? Is that what he has caught there? Do you see that?
I do see that, and that might be an amphipod that's living upon the Solmissus.
Okay.
That's what I would guess. Oh, that was just fantastic. Thanks a lot. And that's before the transect even started. A bonus Solmissus.
Here's something. Some schmutz. Yep, whenever you are.
Success?
I don't know. Now.
Great.
So that's sample 6. 6W. Okay.
Yep, that was the 6th sample. We still have room for 3 more organismal samples, and we have two transects to get them.
Yep. There might be a little more life down here in the water column. It looks like we've been seeing a couple more diverse things than up in the shallower spots.
And the light seems to work better. It's darker down here. I am disconnected.
These cating gnats are everywhere. They're just in all the whole column, basically.
Yeah, they got a good thing going.
Yeah.
Okay, watch leads and shoreside. This is the start of the 900-meter transect.
Thank you.
1703 UTC.
So this is transect 5.
This is the 5th transect.
Got something pink at, looks like a shrimp maybe, at 4. Just off screen, going off screen right now. There it is. There we go.
Oh yeah, another suggested. It is on the move. Really nice.
There's something at 6 o'clock. There we go. What do you got there? Looks like a little UFO.
It does. Does that look like that doliolid again?
Another copepod in front. Ooh, got stuff flying by.
Maybe it was a siphonophore bract. It was hard to tell.
More of those forams. I got something at 6. Oh, never mind. Go ahead. There we go. Tina 4.
Yeah, we have here, this is Burrowie. It's a ctenophore without tentacles, without lobes. It's basically just a giant mouth. And I shouldn't say just a giant mouth, but it has a giant mouth, and it is very good at engulfing prey. It's swimming forward. Its mouth is out front there. Can just see it. And sometimes they eat by breaking off part of a soft gelatinous organism, or sometimes they just can eat the entire organism, and their front end of their bodies are really, they can really extend far and engulf larger prey than themselves even.
They're stretchy.
They're very stretchy. That's a good way to put it.
Here's a Medusa maybe, an off screen?
Might have been.
We have Medusa at 1 o'clock. There you go. Yep, there you go.
Oh, you are good. Good job, Steph.
Oh, cool.
Ooh.
Now,
I
this
like
looks
this
like
guy.
one of those little jellyfish that usually has the red insides.
Yeah, it does. It looks like it's a coronate jelly. It looks like it's just not used to being able to see inside so well. So it's paraphila. Really, really. Yeah, I like this shot a lot.
Maybe it would be red if we got close to
You can
it.
see it's 8. I think it is red. The gut is red, but you can see it's 8 gonads really clearly in this shot.
Right,
They're
so
sort of-
this is really speaking to how red disappears
Yeah.
first in the water column. So he's actually pretty far away from us.
Yeah.
And if we got close enough, you'd actually be able to see his red guts coming.
Yeah. You can
When the light increases, you can start seeing the red interior. And that red covers up what he's been eating. There he goes, his red. So that was really neat.
Yeah, it's
Because
fantastic.
from far away, he looked totally, like, he looked the same color as the background.
Yeah.
And when he's up close, he's this bright red color.
Just great footage. And it's the white parts that are the gonads there. Sort of, there's 8 of them in pairs of 4 pairs. Oh yeah, you got that animal from lots of different angles. Really beautiful. There's just a single species in that genus, paraphila, and it's all over the world. In the ocean, not in the rainforest.
Yeah, I don't think it'd be much use of hanging out from a tree.
right, so we've gotten some really nice views of things already at this depth.
Little fishy.
Nice little fish. Do not know what it is, but hopefully someone will. Yes. Running the zoom has got to be a real challenge in this. Hats off.
I know, I think I probably would have tapped out of that job 3 hours ago.
Yeah.
That's something center screen, maybe?
Nice.
Oh, neat. Look at him.
Oh, yeah. We've got the same paraphila again.
Nice.
See if we can see anything living on it or if it's caught anything. That's really beautiful view of that's the mouth area. You can see the 4 little white dots around the outside. Those are its sensory organs, Ropalia.
I like how it puts its tentacles in front of its bell.
Yeah, it's got its tentacles out in front of it because it's that's how it hunts. It's a cruiser. So a lot of these jellies are, sit and wait predators, and this has got a different strategy. It's cruising through the water with its tentacles out in front of it when it encounter prey actively. And it has the dark red gut, presumably to mask bioluminescent prey that it ingests. So it doesn't want to have a bright flashlight in its
stomach.
In its stomach, exactly. So that red gut covers it up. Yep, we've definitely found some.
Ooh. Yep.
Thank you.
Copy. Okay. Let's speed up.
Keep us on track. So I know George is hoping we'll turn up a relative of paraphila. Unusual form of atolla. But
Is it a similar body shape, or am I looking for something like way different?
It would be more spread out,
Okay.
less conical.
Okay. Still with the red guts? Or we don't know?
Almost certainly with the
Okay.
red guts, but with lots of tentacles around the outside.
Okay.
Well,
Ooh, what was that?
that was
Shrimp or something? Something red just shot by. We got something long at 4 o'clock below screen. I think it's probably gone now. Yeah. Oh, there it is. Whoop.
well, whatever it was.
I don't know what it was.
That's okay.
That's something dead ahead.
There's always more.
Is that forearm? Oh, that's different.
Oh no, see, that's a larvacean there.
Neat.
And you can see the tail of the larvacean is beating, and that's what creates the water current that it flows through its mucus house, which acts as a filter, so it can.
Does it catch all of its food on its house?
Yeah, it actually separates larger particles that it doesn't want to eat through the filter house, and then it gets the ones that it does want to eat and
Come through.
on an inner filter.
Nice. Got something? Yep.
All right, you guys are dialed in now.
What am I looking at?
I think that is hoop-shaped
Sea snot.
schmutz. Yeah. That was really... Somebody
Really
blew a
well-formed.
smoke ring.
Yeah, right?
I gotta remember that we
Something at 4?
want one of those larvaceans.
Oh, did we? Okay.
So it would be
Collect one.
good to collect if we...
Probably a Cadignath?
Yeah, it's another Cadignath. This one seems to have more pigment to it. So I presume it's different than what we saw before. Also don't see the prominent gonads in the tail region of that guy.
Oop, Siphonophore?
Oh, Medusa went by. This is fun. Okay.
Ooh, ooh, ooh. What was that?
On the left.
Yeah.
Was a worm or
I
something,
think
right?
so. Here's
Look
another Medusa going by.
yeah? Great. I would love to see one of these pelagic worms.
Yeah, let's see.
There are scale worms out here, and there are some other worms with some fantastic eyes.
Something swimming by.
Yep. You can really tell, how much looking it takes
Yeah.
to gather the quality data that we're interested in with the good video, or even the collecting.
Something at noon. Maybe Cadignath. Ooh. It's a little... Yeah. Pyrosome. What is this?
No, I think this is Paraphila again.
Paraphila.
It's very nice. Oh,
There's
there's
two of them.
another. So we're still not quite halfway through this transect, so it's got lots of time to find
Yeah,
more.
something red dead center screen. Shrimp maybe.
Yep.
Little red shrimp. He's neat.
Yeah, so
Cool.
he does not look like a suggested, right? Because it doesn't have the bent over.
He has the broken back though.
That's enough to solve it, I think.
Does it Nathaphausia?
I think. I'm not sure.
Uh-huh.
They hold their antennae like that. I don't see the projections off the back of the carapace that you usually see on them. So I'm not positive about that.
That was
You
great
get
footage
really
of it.
red like that.
Here's another ring. I think that's probably snot. Another one, maybe?
See. How many thousands of organisms do you think we've
Like not seen
motored
it all.
through?
Yeah,
Yeah, well, there's lots that we haven't seen at all,
Yeah.
but a lot that we have just barely been able to see because
This
they're
at
small.
6?
I think that was.
Oh, just because the lights can't pick them up, or we can't see them well.
Yeah, or we
Camera can't see them.
just see them a tiny bit, or they're just on the edge of us. It's
It's teeny tiny.
so many.
Looks like maybe
Thinking of
siphonophore. Here we go.
siphonophore, yeah. And
Excuse
it
me.
says, "I've had enough of you."
Bye. And Kiddicknath, maybe? 9? Very shiny. It might have been a fish.
Yeah. That's what I was thinking, but
Here's something. There we go. There's another one of your ctenophores.
shoot. Yes, this is the way it goes.
Ooh, that's different.
Oh yeah, so that
That looks
is
like
a
it.
Physonex siphonophore. You see, it's
Siphonophore,
got
yeah.
the float on the top, and then it's got the swimming bells, and then what's it feeding on? Oh, look, can we zoom in on the? I think it's collected food. Are those?
My gosh, they
Are
look
those
like
medusae? Are those
They look-
atollas? I mean, paraphilus? Yeah. I think those are paraphilus. So we are seeing here some nice, fantastic example of feeding interactions. I love it. Okay, that was great. Oh no. George says those were gastrozoids. I thought they were little tiny jellies. Well, the gastrozoids are the feeding zoids on the siphonophore. Which George identifies as apolemia. Does that look like? Oh, we'll have to review what those were.
Let me get- There's something there at 6 o'clock. What do we got here? Another siphonophore?
Yep.
Sweet. Yeah, this one has the same little red.
It does, but this is a different siphonophore altogether. This just has the 2 swimming bells.
Mm-hmm.
Oh. Now, did those look the same? What was connected to them? Like the same?
It looked like the same, like that same texture, same color, but the little, I don't know what I would call like ring of tentacles on the outside didn't look the same. All right, so. So yeah, we didn't get enough footage of it to really get a good idea.
Why? I hope we find another.
Oh, shrimp or something. Mm-hmm. Yeah. Mm-hmm.
There we go.
Mm-hmm.
Oh, there we got a squids guy. Squid phone, please. Oh, that is great.
Oh, no way.
Hello.
Go ahead, Mike.
Mastigotoothed squid.
Which kind?
Yes, that's a mastigotoothed, and I have to get a little bit closer to tell which genus it is, and then I have to look it up. But it's one genus has recently been split up into a bunch of different genera. But what you see there, it's got 2 long ventral arms, and then dangling down, the white dangling down is the tentacles, and it holds the tentacles in the arms because there's almost no musculature in them. So they drift along like that, and the tentacles have microscopic suckers on them, and they're very sticky. So if something bumps into them, then they whatever bumps into them basically sticks to them, and then it will try to get whatever it is up towards the arm crown, where it can hold it and bite it into pieces and swallow it.
So is it eating fish?
I think the species there, well, I don't think it's eating anything right now, but
yeah.
They eat, when I've opened stomachs on them, I found small crustaceans and that sort of thing. I think that's what they typically feed on. And they drift fairly close to the bottom usually, but sometimes you find them up in the water column as well.
Thank you.
I think the species, it used to be called Mastigoteuthis magna, and I think it's now Magnoteuthis magna. But I have to actually look it up to be sure about that. I'll put it in the chat.
Thanks, Mike. That was great. Mastigoteuthis.
Yeah. Yeah, except I think now it's Magnoteuthis.
Oh, Magnoteuthis.
Yeah, I'll put it in the chat.
Great job on that video.
Mm-hmm. Yeah, we got something right there.
Oh, nice. Now is that
fish?
What I didn't think before. That's a snipe eel, right?
Oh, look at his little head.
Yes, that's a snipe eel.
Oh my gosh, he looks like the dog from Nightmare Before Christmas, the ghost dog.
Oh, awesome! The ghost dog. Yeah, he swims along with his mouth open. You can see that.
Look at the mouth. Yeah, if you look at the mouth, you'll see it doesn't close all the way, and it's got really tiny teeth that are pointed backwards. They swim around and they actually tangle up the antennae of shrimp on those really tiny teeth, and that's how they feed on the shrimp.
Wow, that's great. Great video. Great sighting.
The other thing about snipe eels, a lot of the midwater fishes, particularly the long ones, have a characteristic posture. They'll either be vertical with their head up or vertical with their head down when they stop swimming. The snipe eels will just look like a piece of rope that you tossed in the water, and it just, however it lands is how it floats. It doesn't seem to have a characteristic posture like many of the other midwater eels do.
Too cool.
Really cool. I think we should just skip down to this depth next time.
Yeah.
Except that it's not the same all the time. So we are in our 900-meter transect. We have already completed 300, 500, 600, 700. And after this 900-meter transect, we are going to do our final transect of the day, about 10 meters off the 10 meters off the bottom, which is at 1,080 meters. So we are basically investigating all the way through the mesopelagic zone, which is 200 or so, 200 meters to 1,000 meters.
Ooh, this flashing thing.
Yeah. Can't see it anymore. I lost it.
Got something that, yep.
It's another one of the paraphilas.
Yeah. Oh.
Ooh, another
Siphonophore?
siphonophore. This looks like what we called apolemia before. Can we get focus on the... I'd love to see those red gastrozoids up close if we can. That's really, really neat. So... Now that's... I thought I was eating them before, but no, George Matsumoto said that those are basically the stomach zoids, the gastrozoids of this animal. So those are the...
Looks like a bit of Christmas garland.
Yeah. So you can see it has all those large... the white parts, it will stretch way out into the water column, and that's what does the catching of the prey. And then those big red gastrozoids, I guess that's where the eating goes on.
Yeah.
That was really cool.
Ooh. Schmutz. I'm good at finding schmutz.
Yeah. Yeah. I'm surprised
You saw him before? Okay.
how variable the paraphila is and its colors.
There we go. There's a little X chromosome again. Nice.
Yeah.
Is that a different type of foraminifera? Radiolaria?
This is what I thought was a Phaedarian, although that one looks a little mucky, but
And there's something shiny. It's up to you.
go for the jelly.
That's another one of those, right? Medusa? Narcomedusa?
It is another Narcomedusa. Very nice. And this is Solmissus again, I believe. And this is another one that holds its tentacles out in front of it as it cruises through the water. It's really nice. You can see its small mouth in the central. That's on the opposite side of the animal. And
Looks like that film inside is really shiny. Is that what I'm saying? So it's reflecting the lights off the ROV. The blues.
Oh, yeah. I don't know what that's about, but you can also see the stomach pouches there associated with each one of those tentacles, and that's part of how we tell apart the different Narcomedusae. It has to do with their stomach pouches and how they are in relation to the tentacles. And
Distinguishing characteristic.
yes, at one time they broke up the families that way. That turns out to not be the best way, but it's still, they do help you tell one animal another apart from another. All right, that was fun. Is that another paraphil up there?
Not sure what that was.
I think so. I hit those bright. All right.
There's something. At noon.
That's our larvacean house. We wanted to collect one, so can we
grab
make
'em?
this collection? That'd be great. Thanks.
So sample 7.
Yes, that's what I'm on. And this was requested by Russ Hopcroft from the University of Alaska.
Sample 7B.
This would go in canister 3. Oh.
Oh.
Yeah, I think maybe we better not, because the animal will probably swim away if the house busts. Thank you, though.
Shrimp.
I had that distraction.
Another shrimp and Okeanos.
Yeah. Yeah, it was a suggested, the shrimp.
There's something new. 6? No, it's close. Too close. What I was looking at, but yeah, go for it. Ooh, might be a worm.
Ooh, let's see that.
Ooh. Yeah, worm.
Yeah, that's looks like Tomopterus. Well, they're very fast.
That's a polychaete.
It is a polychaete, yes.
Okay, this is the end of the 900-meter transect.
Thank you.
Transect.
So we probably ought to put that worm in at the family level, Tomopteridae.
I'm coming after you after this and having you help me fill out all the taxonomic information.
And we could send a frame grab to Karen Osborne at the Smithsonian, who might be able to, or maybe a little bit of the video.
So now we're going to drop. How deep is it here? 1,000?
It's not as deep as where we started. It's not 1080 anymore. I think it's just over 1,000, and so we're still going to go down to 10 meters above the seafloor and do a transect there. And we will collect our final water, and we still have 3 canisters if we want to use them for additional sample collections. So it's 7?
7, that was 7, yeah. Sample 7, right?
We're gonna do sample 7, right?
No, let me check. We were gonna do 7.
Yeah.
And it was a fail, so we're gonna. The next one will be 7. Mm-hmm.
Are you looking for me?
Mm-hmm. The chinks. It looks like the snow's increasing. Isn't the marine snow really sparkly?
It does look very sparkly as we descend closer to the bottom, out on the eastern edge of the Blake Plateau.
Yep.
So if you're just stepping in, we're getting ready for our final transect of the day, where we've been exploring the water column over the Blake Plateau, out on the eastern edge of the Blake Plateau, about 170 miles from Florida, Georgia, both. Yeah.
Offshore.
Southern United States.
Mm-hmm.
And we've been slowly working our way down through the water column doing transects. We've already completed 5 transects, and this will be the 6th transect of the day. And just prior to this transect, we'll be making our a water collection for environmental DNA, so that we can basically sequence the DNA that's in the water that comes from either loose DNA or sloughed off cells or gametes that are in the water. And then that will provide another means by which we can characterize what's in the water at these different depths.
Local population.
Yeah. And that could capture things that we're not physically able to see, or it may capture things that we did see.
All right, about 40 meters above bottom right now. Yeah, it's something gray at 4 o'clock. Disappeared now. Looked like a big fish, but it would have been pretty far away. It's very disorienting. I have nowhere to. I don't understand what I'm really looking at. It's just blue.
Yeah, it's
so I don't really
really
have
hard.
Yeah, I don't really have like a point of reference. You're about 25 meters above bottom right now.
And it's looking like we will have a little time to look at the bottom.
Yeah, that'd be good. Get an idea what's underneath us.
Yeah. I feel like I can see sponges on the bottom there. Is that real?
No, I
Is
think
that
that's
reflection?
the reflection of the camera housing.
Wait a second.
And how
Those are sponge flares.
sponge flares.
Potato.
We were watching Ghostbusters last night, and Dan Aykroyd has a line where he said, "I watched the only documented mass sponge migration." I got such a kick out of that.
Yeah, they do move.
There's somebody. Right, about 20 meters up.
Yeah. Yep, about 20 meters off the bottom. You notice Mike didn't even ask us to try to catch that squid.
I don't know if the squid would have waited for us to...
No, plus I think it was
Too big, maybe.
maybe comparatively well known.
It's a little blackfish.
Yeah, we've got a lot of those.
Yeah.
And you'd be surprised what you can catch with the sub when you're down deeper than 500 meters.
Mm-hmm.
I've caught fairly big squids in suction samplers.
Oh, really?
Yeah, good to know.
Yeah.
So we are just about at 1,000 meters.
Yep.
Oh,
Yeah.
there. All the light is now gone.
Yeah. But we're at 15 meters up. I think I'm seeing bottom now, but I could be. Yeah, see the dark spots? It looks like sand, yeah. Sandy bottom.
All right, well. Ooh. So a lot of pelagic animals actually live fairly close to the bottom and in association with the bottom.
There's something black right underneath at 6 o'clock.
Yep.
Not only are there pelagic animals close to the bottom, but also the vertical migrators. Some of them come down this far, so they sort of compress when they get close to the bottom, even though they're not used to having a bottom around. Oceanic vertical migrators essentially don't know what a bottom is, but if they get over a depth that's shallower than what their daytime depth would normally be, then they compress near the bottom and they start interacting with bottom fauna, which is good for the bottom fauna and bad for the pelagic animals.
Hey.
Bentopelagic.
Ooh, a red side dipid. Goodbye.
We've seen a bunch of them.
Yes.
Yes, please.
So this is 7W.
I want to see that red sidipid. Might be Vampiroctina.
There's another ctenophore right in front of you. Yeah? Right here?
Yeah.
Right in the front.
Let's take care of the water sample. Having a lot of fun.
There you go, look at that.
Oh, yes. Very pretty. One of the deep red sidipid ctenophores was recently described. For a long time it was, well, it's had common name Tortugas red. Mm-hmm. And it was recently finally formally described, but it turns out there's other deep red sidipids. So
Oh, what's that? Oh, a shrimp. Did you guys fire the Niskin yet? It didn't close. Okay. They fired it, but it didn't actually shut.
okay.
I'm not sure. You're muted.
I think we got another lobe 1804. So is it started, or is this just a bonus? This is the bonus. Yes, it's always a bonus animal. I think this is Bathocyroe, which is a lobe 1804 that might start flapping away. It has this very distinctive swimming motion when it's disturbed, almost like a leapfrogging. Yeah, I didn't. So our final eDNA collection it's not successful. The Niskin bottle opened, but it did not close back up like it was expected to, as we hoped it to do. So we're going to have to do without that sample. I'm going to make up for it with three specimen collections at this. I guess so. I want to see it. Yeah. Boy, I like this near the bottom. Yeah. Yeah, so I think that's a red sedipid in the, way there. Yeah. Oh my gosh, this is so much fun. Oh, me too. 3 days of missing. Yeah. That's why I was up at 4:45 this morning. I was like, yes, it's nice out. to you. Oh. Oh. A fish, I think.
Is it fishy?
Oh, what was that?
What was that?
I don't know.
Okay, watch these and shoreside. This is the start of the last transect, 10 meters off bottom, starting 1803 UTC.
Thank you.
1,006 meters. All right. All right, zoom. Tina 4. Oh, there's a big fish in Sirius's camera. There we go. Look at Tina 4.
Oh, there we go. Oh, I love this. So,
Look how cool.
this is a side dipid.
It looks like he ate a butterfly.
Yes.
Isn't that pretty?
So I don't. Boy, I'm trying to think. I kind of want to collect that.
I mean, yeah, why not? If they have time. Is it too late?
Is it? Well, we may see it again.
I think so. I think we saw 5 or 6 of them already, yeah?
Yes. Thank you. Turning into quite the Tina 4 dive.
Mm-hmm.
Just because I don't think that is. I don't think that's the recently described red side dipid, the Vampiroctina. This is very likely an undescribed species, he says. Not really being a Tina 4 guy, but I don't think this is described. Let's see what George has to say.
So 2107, dive 3. This will be sample 7 because the water was a blowout, so if we get it. Ooh, ooh, ooh.
Did it go?
Yay!
Fantastic.
He's in number 3. Suction bucket 3.
Wonderful.
Sample 7.
This has turned into quite the Tina 4. Well, it's the way it is. No. Thanks for that collection. That was great. It would be terrific if we come upon that same animal to try to get some more video footage of it.
There's something.
Oh, no, that's a different jelly. That's a Narcomedusa. Oh, for sure. Oh,
There's something red at 9 o'clock. Oh. 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 There you go. Dark spot, yeah. Another one?
yeah, I think that's that same deep, dark red sidedipid.
Another one here.
Yeah, it's common near the bottom here.
So we have, what, 2 buckets left? Okay.
Yeah.
Mm-hmm. Ooh.
Oh, nice video.
Now, is that the same species we just collected? Do you think that's something different? It looks different.
I think it's the same.
Same?
Yeah, it has tentacle sheaths. So it can pull them all the way inside the edge of its body.
Look how cool. And this is the Vampiro one, you said?
No, it's undescribed.
Undescribed. Look how beautiful.
It is.
There's a little green and red, maybe even purple in there.
Yeah.
Some pretty good Halloween colors.
Yeah.
Cool.
So Vampirocteuthis is narrower near its mouth end than that guy. I think its tentacles are still a little red. You see, the tentacles on that one are so bright white.
Mm-hmm. Here's another one.
Yeah. Really nice. So I'm hoping we come across that little Narcomedusa with 4 tentacles.
Mm-hmm. Here's something. That's schmutz.
Yeah.
Okay.
Good eyes.
We don't need to stop for schmutz. Another one.
There's that guy. He's got his tentacles deployed.
Oh, look how beautiful.
Really nice.
So what's happening on their combs that makes them so light? Are they actually creating their own light? They're reflecting
No.
light.
They may be able to be. They may be bioluminescent, but that's not what we're seeing. It's just that the combs refract the light, and so it splits it up into a different
It's almost like a hologram.
wavelengths.
Like a hologram on your, like a plastic hologram. I don't know what the...
Prism kind of
Yeah,
thing?
yeah.
Yeah, so it just separates it into the... Ooh, look at that! Now,
They're buddies.
They do not look the same. I guess they are the same.
This one's more orange,
No,
smaller.
they might not be the same guy. Wow. Okay, now I'm really confused. I like that. My normal state of being. They are. They're so abundant. All right. Next midwater dive is 10 feet off the bottom for 8 hours.
Right.
That's
Yeah, good call. Picking the 10-meter transect off the bottom.
Here we go. Something neat at 9 o'clock.
good eyes.
Same thing,
Oh
yeah.
my goodness.
That one looks different now.
Oh, it is different. It's got tentacles or appendages up front. Oh, that's definitely not the same.
He's not nearly as clear. He's more opaque.
And does it have side branches on its tentacles?
Yep, looks like it. 4 of them, maybe?
No, but on the main tentacles, do you see any side branches? I don't.
No.
That's a collect.
Collect this guy?
This Tinoceros? Never seen this guy before. George Matsumoto says it's Tinoceros.
Oh, wow. It's amazing.
All right, so I've never seen that. But
This is going to be 8B.
Yeah, this will be 8B. We're going to use all our Rain-X up.
Mm-hmm.
It's tentacles. Yeah. But did he get in? Thank you. All right, so. Huh. Tinoceros.
That's something else.
Like rhinoceros. That was a name given by Rich Harbison.
Yep. Looks like a lanternfish or something. Nice. Dragon net?
I don't know.
There's another one over here. Kind of mucky down here. There it is. Yep. No, that one looks different. Look at him.
He is cool. It's a good video, though. I think someone will be able to...
Sit still. Stop running away. Mm. Schmutz, I think. So we keep seeing the same shape.
Yeah.
And there's nobody in it. There's always, sea snot. There's another one of those red ctenophores at 6.
Hopefully it can get some more.
That's different. Ooh.
Yeah, there's Bathyroe. That's a lobate ctenophore. You can see its auricles, this looks sort of appendages inside they're sitting in one place, not waving around. Some of them wave them around.
Mm-hmm.
That was nice.
There's something shiny. That's probably the... Yeah. Yep, ctenophore. Another red ctenophore.
Yeah. So I haven't seen as many medusae down here, but...
Shrimp, maybe? Another one of those.
That's a
Lobe.
Bathyroe again. Really nice.
Neat. There's something long underneath him. If you go back to the back to... Yep. Oh, same ctenophore. They are everywhere.
They really are.
There we go, something at noon.
Oh, there's a Narcomedusa, like an Aegina type, or Bathychorus. Could make our last collect.
Yeah, this might be the last collection.
Yeah, you want it? Shall we make it now?
Yeah, go for it. That's what you...
Yeah, if you can collect it, we'll take it.
8 started at 1814.
Oh.
Oh.
Oh, it's so pretty. It's got a little
horns.
It's got a hitch in its...
Mm-hmm. There he goes.
Oh, wow. That's great. Is that 9?
Yes.
And canister 1. Agena. For now. Thank you for that collection.
What was 8? What did you give the name of 8? I have Tenopterus or something?
Tenoceros. C-T-E-N-O. Oh. Ooh, now we got a fish.
Ooh.
Good fish. That was cool.
Correct my spelling.
That was a shark.
Yeah.
That was really neat.
I've got something at 9 o'clock.
Is that
a
a
dark spot? Fish, never mind. He's gone. I think there's another red Ctenophora.
2 of them, yeah.
There's a Narcomedusa or something at noon or center screen.
Yeah, I think that's the same one we just collected.
Okay.
It's really nice. Come on, come on.
There you
Beautiful,
go.
beautiful to have this footage to go along with the animal. Oh, who was that just went by? Yellow.
I thought it was another Ctenophora, but
There's that Narcomedusa again.
Mm-hmm.
Thanks, this will be great to have.
This one's upside down.
Yeah.
Pretty chill.
Look how wide open it's holding its mouth there.
Looks like a bowl.
It does with the 4 legs.
It's waiting for someone to put Halloween candy in it.
Yes. Oh, there's nice swimming. Well, I think this is Bathychorus. You see these small little tentacles, the tiny ones that are on the edge of where it's...
At the bell part?
Yeah.
Like it's...
So I think that rather than Agena, then that would be Bathychorus. So already put the wrong name in the books, but we'll fix it. But that's why the video is so great to have. Thanks very much, video. Ooh. Another nice Ketognath arrowworm. I think that's Bathysiroe up there. You can see it using its lobes to swim, reorient itself in the water column. Just gorgeous. Wow, that is great. I think that's just spectacular with the refracting light off the comb rows. Yeah. What else is out there? So if you're tuning in late, we are out on the Blake Plateau, NOAA Ocean Exploration, doing Midwater Transects. And this is our final transect of the day out on the Blake Plateau, on the eastern edge of the Blake Plateau, about 180 miles off the coast of the southeast United States. And we are 10 meters off the bottom, conducting our final transect of the day. Great. That's another beautiful view of Bathysiroe. Another one of the deep red sidipid ctenophores that we sampled earlier. Likely an undescribed species. Zooming in on another comb jelly. The comb jellies have been everywhere on this on this midwater dive. This is Bathysiroe again. You can see those ciliated auricles inside the lobes of that lobate ctenophore. Looking, I think that was a siphonophore, but it never quite came into focus. Okay, that's really interesting. That is, oh my gosh, I think I've seen this before. This is something we haven't seen so far in this dive, though. So if we can get some good view of that. So you can see that it has maybe this is a larval. It has the tentacle still, but maybe this is what is the larval form of a lobate ctenophore. Oh no, George. Oh no, yeah. George Matsumoto was wondering if it could be Lampeia, or also that it might be a larval lobate ctenophore. So these lobate ctenophores that don't have the tentacles, they do grow through a sidipid stage with the tentacles. Yeah. There's a lot of... Yeah. And that one you could see the small lobes forming already, so that was almost certainly a lobate ctenophore on its way to growing up. So it still had the tentacles that it fully loses as an adult. I am learning an awful lot today. I'm enjoying it. There's nothing better than learning, even at the age of 56. So never stop. Yeah. Welcome back. We just got some great footage of one of the lobate ctenophores, and then
nice.
right afterwards the lobate, like a larval form of the lobate.
We saw him earlier.
Yeah, we saw this fish before. This is an interesting looking guy. I like how it's just delicately flapping its fins there every once in a while.
It's weird looking, isn't it?
I missed part of seeing a fish like this before. This looks like a melamphid to me, or is it a bathylagon?
I have no idea. Melon, what'd you call it?
Melamphid. But I think this is an owlfish, so a bathylagon. But again, my picture's not all that clear right now.
Got some more water column fish up here.
Melamphid.
All right, gonna change pilots.
That could have been a dragonfish off in the distance to the left.
Okay. What is that?
A large siphonophore down there.
Maybe.
So we have a pilot change.
Yeah. There's another fish around 3 o'clock.
Another one of those red sidipids.
Mm-hmm.
Oh, yeah.
I don't know if anyone said this before. This is about fish. Oh, out.
You're cutting out. Can you repeat?
I was going to say bathy laggards tend to be more vertical in the water column hanging out, whereas like dragonfish tend to, in my limited experience, tend to be more horizontal.
Okay.
So I think that last dark fish that went by was a bathy laggard.
Okay. Thank you. I don't know what we have at 6.
Quiet.
That's neat.
Wait.
Ooh. Tina 4.
Yeah, that's the ctenophore. I think that's Bathyroe again. And I think, and I misspoke before, that they do still have tentacles. They're just they hold them inside the lobes near the mouth.
They're tucked.
Yeah. There are some lobates that have completely lost the tentacles, but most not.
We got something at 6 o'clock.
It's another Bathyroe, yeah, lobate ctenophore. So we must have about 10 minutes or so.
Yeah, another one.
Yep. That guy has proven to be very common at during this transect.
We got one center screen. All right, right at noon. There you go, that guy. Mm-hmm. That one looks a little different.
I think that's the same again.
Same species.
Yeah.
They're super cool. So these and then the red ones.
Hey, there's two kinds of red ones, at least.
Yeah, here's it looks like another red one here at 3.
Yeah.
And 4.
Oh, yeah.
There's 3 of them, yeah. 3 of them right there. Yeah.
Yeah, look how many.
Look how long his tentacles are, man. Cool.
Thanks. That one that was dead center adjacent to those other two, it looked a little different.
Mm-hmm.
But I don't know. Ooh, there's that Bathychorus Narcomedusa again. Let's see if I'm missing anything in the chat room. No, just me lamenting my lack of knowledge.
Oh, it's better than mine. I'm like at a loss out here.
My lack of knowledge is way better than yours. That's okay, though.
You can't know everything.
Nope.
Another low bait ctenophore, maybe? Yep, it's 6 o'clock right now, about dead center. Should be the same one we've been seeing. Yeah.
It looks like a ghost.
It does.
Very Halloween-y.
It is.
A ghostly.
It's clear. I love the little combs.
Yeah.
They really look like UFOs. Like little lights like flicking off.
Really do. This has been dominating. We've seen the red ctenophores, the red cydipids, the Bathycyroe, Bathychorus a lot. That melamphid fish, we saw that twice, right? At this.
Mm-hmm. The owlfish. She
Yeah.
said no. We saw one at a deep, shallower depth, but I don't think we got a name for it when we blew past it that time because it wasn't on the screen long enough.
Ah, well.
I think you got a little red shrimp coming up. Oh, yeah. There he is.
Fantastic.
Cute. His big
got-
orange eyes.
My gosh, that's so.
They look like fins, right? Not
Yeah.
like a.
He looks like he has a mask on.
Yeah, right.
That's really cool.
Mm-hmm.
I don't think I've ever seen him from that
In the
from
front.
that angle.
Looking at you.
Yeah. So, antennal scales.
Neat. Oh yeah, his little clear eye mask.
Yeah.
Yeah, what is that? Another low bait ctenophore. I don't know how much you're going to see.
Yeah, and that's Narco up there. That same Bathychorus. How about the jelly this time? The Medusa, yeah. Yes,
He's neat.
really is. Great footage.
Which one did you say this is, Alan? Sorry.
This is Bathychorus, so it's similar to Aegina, but you can see these right along the bell margin where the velum connects to the bell margin. There's really tiny tentacles, 4 of them, one on each quadrant.
Oh yeah, I can see.
Yeah, and those.
That's what.
So that's
Venus.
like. That tells us we're either Bathychorus or Siggywedelia, something like that, but this one is Bathychorus.
Okay. Cool. I will try to remember that, as opposed to saying Aegina.
No worries.
One of those red ctenophores.
Yeah. Nice.
Sorry, what pilot?
Oh,
Okay.
sounds great.
Far.
Ooh, hello.
Ah.
Oops.
Oh,
Another shrimpy.
nice little shrimp.
Hello.
Suggested. That's just great. Cruising through the water there.
You're done with his vest on him, right? Go, come here.
Come again?
Here, Pico. Pico, come here.
I couldn't quite hear you, Mike.
Might have a bad signal.
I think so.
What is that?
That's a Keating now.
Yeah.
Right? Oh, no.
Yeah.
Yeah, it is. Yeah.
He's just not moving.
Yeah.
Maybe he's a corpse. Or just frozen and
Did
out
that-
of pure terror.
Yeah. Did that conclude the transect? That was great. So I think now we have some time for some bottom
You want to
exploration.
check out the bottom real quick? We can do that.
I would love to.
Mm-hmm. Looks like it's going to be mostly sand.
Yeah, it might be. Be interesting to see what we see if it's all sandy bottom.
Oh, I got a couple of eels or something. The bottom looks the sand waves.
So should I shift the quad split over to the normal ROV bottom mode?
Yeah, probably rolling for a few minutes. Looks like a Thalassia detritus. Geez, we're far away from land for that. No, just find out. Yeah, just look at the habitat and you know what we were diving above. Maybe get a good zoom on these eels. Trying to figure out what they are. Any life you see, really. It's gonna be pretty barren. I think that's just rocks. Yeah. or Detroit. You know, that looks like dead sargassum. But see the eels in the far. There's 2 ahead of us. Yeah, that's cool. A cutthroat, maybe. That's good, thank you. Looks like there's a few of those ahead. Maybe 3 or 4. There might be a hydrate here sticking out of the bottom at 5 o'clock. Right about 6 now. There you go, center screen. Oh, wow. It's something. Yeah, it might be Stichopathes or something similar. There's something white in center screen too. It looks like it could be a shell. Maybe a snail or just above the Stichopathes.
We are on the bottom.
Yeah, above that. Whoop. Yeah, I'm sorry, if you back out a little bit, just go wide. It's actually on the sediment behind that, above it. Yeah, the shell. That.
Oh, yeah, nice.
Yeah, nice snail.
Snail.
Sweet. That's cool. You can see the pteropod shells in the sand. So that's a nice little gastropod there. The black coral? Yeah, I mean, we can zoom on it if you want.
Stichopathes?
I think so.
Maybe?
If you get close, you can see the thin, they almost look solitary, but they're not actually polyps. See the one?
Polyps,
Yeah, the 1 or 2
1 or 2
polyps.
tentacles?
Yeah.
See that little,
The
little fish swimming along
amphipod.
that?
I think it's
Is
amphipod,
that an
isopod.
amphipod right there?
Mm-hmm.
Nice.
So yeah, I would definitely call that a black coral. It's awful yellow for Lutgeni. Lutgeni is usually a gray. So my guess is some type of Stichopathes. Oop, there's a black fish. That guy. Oops, it went away.
Ah, nice.
Yeah, it's mostly sediment, like real loose sandshell hashy, kind of. It creates these like mounds, right? See the water ripples on the bottom. There might be something ahead there at 6. I'm sorry, at 9. There's also something at 9 over here. Yeah, let's go there first.
Nice.
Ooh, look how reflective. Oops.
Ooh, that was nice.
Yeah, it looks like we've got something here at 6 o'clock. Maybe a sea pen. Yeah, some kind of sea pen. That's pretty. You can see the bulb that sits inside the sediment. Okay, and then when you guys are done with this, there's something around noon in the distance. Yeah, probably another sea pen at noon. Probably a different species. So Panatulacea. Yeah, it's a different species. Cool. I have no idea what the species is, but yeah.
Oh. That's a different sea pen.
Mm-hmm.
Interesting with their branches.
Yeah, if you get
The
close
three
to them,
dimensions.
sometimes they'll pop down into the sediment. Look at how weird the sand is. It's like super thick here and then flat.
That's right. That's...
Yeah, we don't have to stay down here very long. It's just nice to see what's here.
Yeah, absolutely.
If you want to go back up into the water column and do another transect, I'm happy to do that too. That's pretty cool. Either way, both.
I'll look for associates.
Mm-hmm.
Yeah, if you're joining in, we have been exploring the water column today, and we did transects all the way down 300 meters, 500 meters, 700 meters, 900 meters, and then 10 meters off the bottom. Now we're spending a little benthic extra time investigating the benthic fauna, the fauna that lives on the bottom beneath the water column that we've been exploring all day.
Yeah, and it's sand down here. We have a couple of fish and some sea pens, which is what we're looking at now. This one's different. It's very furry. Like really fine tentacle, like pinula.
Uh-huh. Did we get a view of the basin there? Was it possible to? Yeah.
We did. You get a view at the bottom?
Yeah, view at the bottom, the base of the sea pen.
Yeah.
The other one was really bulbish, like you could see the bulb it was sitting in. I think this one has that same.
Yeah, I think they saw the base.
Yeah, it's hard to see from this angle.
I was having a little trouble with the chat room catching up to us, but yeah, they've seen what they need to see. They just think it's a very odd Panatulacea, but
yes, that's weird.
Yeah. I can. Well, we have the boxes now.
I think
I'm going to ask the chat room. But.
Yeah. Sometimes if you go close to them, they just shrink into the sediment. There's a possibility if you took your claw out and tried to touch it, it would just boop, go away. So, see what they say.
Yeah, the
collect? Yes. Scott France says yes.
Yes, please.
Go ahead and collect it.
Is the other one nearby still?
Yeah, it's behind us.
Scott said the other one would be even better. Yeah.
This is going to be sample 10.
Oh, yeah.
This one. And then we'll go back. If we can, go back and get the other one.
Just trying to figure out what. So I think, yeah, both would be, both are being requested. Huh.
Hey, Scott, if it pulls itself down into the sediment, do you want us to bother digging him out? I don't know if we'll be able to do that.
Port side box.
No, he says the species won't retract. Okay. Yeah, if you can grab the whole thing, I would imagine. I think he might think this is a new species. Is that the thought process?
He did say it was weird.
Weird. Okay, weird. Yeah. But it shouldn't retract in, so correct. Look at this poor thing. Hey, who chimed in?
Hey, Stephanie, it's Scott calling in.
Hey, Scott.
I thought I should come on live and see if I'm proven wrong about the retraction.
Okay.
You'll have to retract the retraction.
Oh, that's good, yeah. My grad student Upashna Ganguly, who's studying the sea pens, is standing behind me holding her breath.
That's nice. So delicate.
Ooh. Yeah, if it was going to retract, it would have snapped down. But it's pulling its tentacles in, but it's not pulling its whole body underneath. Scott, do we need the peduncle underneath the sediment, or can he snip this off?
You should be able to pull it up pretty easily.
Pull
the
it up.
peduncle would be very helpful. We need to compare that to the rock pens.
Yeah, we prefer to get the peduncle then. Okay.
Yeah, you should be able to just pull it straight up.
Okay. Bloop. There he is.
There you go. So good.
All right, where are we going? Port.
That's a longer peduncle than I was expecting.
Oh, really?
They've really got themselves anchored in there.
Outside. Great work.
Port outer. Thank you. Port outer, Samper 10.
And then...
Yeah, and then there's another one behind us. If it's not too difficult to swing, D2. Hey, Scott, how do you want this preserved? Do you want it in formalin? Or is ethanol good enough?
Yeah, no formalin.
No formalin. Do you need a relaxative? Do we do relaxing?
We don't usually relax them.
Okay. I don't even know if we have menthol on the ship.
We have magnesium
I
chloride.
suppose, you know,
I brought some.
Yeah, if you want to check it out, but so long as you're taking some snips for genetics, because I don't know what the methanol or whatever you use to relax have on genetics later.
Yeah. There was a C-pen.
Yeah, it might be hard to find this other one because you know, just a single stock was pretty small.
Yeah, it might be under you. That's it. 6 o'clock.
And did we have a name on this Penatulacean?
I don't know if they got a good enough look at it. Go ahead, Scott.
Yeah, my first guess was something like a copobelemnon, but we'll see on the close-up here.
Did it disappear? Oh, here it is at 9. So it looks like the tentacles are really on one side. And you can see the peduncle's really bulbous at the bottom. Looks like it might have an isopod with it. Or worm.
Oh yeah, right on the base there.
Got a name, Scott?
So hang on a second. Yeah, what I wrote there first, cophobelemnon or anthotylum, I put it in the chat. It's one of those two genera, we're not sure. But just for people watching and for you, one of the differences between this and the last one we saw is that in the last one we collected, the polyps were born on what we call leaves. There were these big fleshy lobes that came off the primary axis, and the polyps were on that, whereas this one, the polyps are coming directly off of that axial polyp. And as you said, we can see the peduncle really nicely buried. It's got an axial rod, and this looks quite a lot like the rock pens. And one of the big questions is, from what group did the rock pens evolve? And so we're trying to get good samples of the taxa that look like them, that could be them. So that's one of the reasons why we have a little bit of trouble telling you what it is exactly.
Okay.
All right, that's exciting.
Yeah, and I think this will be similar to the last one in terms of collecting. I don't expect it to retract, and it should be able to be pulled up. It's a smaller specimen, so it may be harder to grab.
I'm curious to see that associate that's on the base of this guy as well.
Yeah, I missed that, Alan. I know up near the Gulf of St. Lawrence there are some sea pens. I'm forgetting what genus right now. I don't know if it's Helipterus or something else, but there are fish that lay their eggs on them. And then there are others that have worm associates, polynoid associates.
Yeah.
And then I wouldn't be surprised if there's various crustaceans that are hanging around.
Yeah, we didn't get a really good look at it, but it looked like it might be a polynoid. We shall see.
Yeah, it's really sandy. And you should be able to get the bulb he's sitting in. Like he's got a peduncle bulb underneath him that's buried.
Anthotillum.
Anthotillum?
And
Not cophobelinum?
Nice.
Is he in there?
I think it's there.
It's hard to see what's going in the box.
It is hard to see.
I think it went in. There, I see it. Yeah. I think it's free. I saw the sand in the sand cloud. I think it's free. I think I see a piece in the box. Yeah. Yeah, I see a small piece in the box.
All right.
Thank you. So that's end of sample 11. All right, Alan, what's next? We got, what, half an hour?
Yeah.
You want to go back up in the water column? You want to stay on the sediment? I'm happy to go back up in the water column.
Yeah. Then.
Yeah, 30 minutes, right?
I think we should go back to where the squid was.
Go back to where the squid was. That's what Mike said. Did you GPS tag him? I don't know how to how to get back to him. He's probably long gone.
Those are great collections. Yeah, I think we should go back up since it's a water column kind of day.
Mm-hmm. I'm gonna stow this arm and get ready to move again.
Yeah. So do you want to go just above the bottom again?
I don't know. That's your call, man.
All right.
It's your call. You make the choice.
You got it. I'm gonna.
Okay.
Yeah. How about...
And jump back up to 10 meters up?
I'm thinking like 25 meters.
25 meters?
Yeah, let's get a little farther from the bottom since it's just free time and look around and see if we're still seeing all the same things that we saw a lot of at 10 meters above the bottom.
Sounds good to me.
I'd be curious how sharp that zonation is, if at all.
Mm-hmm. Yeah.
Yes, please, if that would work. Just 25 meters off the bottom. So we have been exploring the water column today, and we've done a series of timed transects at 300 meters, then 500 meters, then in the deep scattering layer at 600 meters. Then we did a transect at 700 meters, a transect at 900 meters, and then a transect 10 meters from the bottom. And then we spent a little bit of time looking around to see what our bottom was like, and now we're picking back up off the bottom. Going to head for 25 meters off the bottom and see how similar that is to what we saw very close at 10 meters to the bottom. At 10 meters above the bottom, we are seeing red sidipid ctenophores, one of which you can see going by. A lot of Bathysiroe, which is a lobate ctenophore. A four-tentacled narcomedusa, Bathychorus. And what else were we seeing down there? There were two different red sidipid ctenophores.
I remember the owlfish.
Yes, not an owlfish though, says the says the chat. I meant to tell you that. Where was that? Ridgehead or a big scale?
A big scale, man.
Yeah, I think a big scale.
All right, we're going to stop around 25 meters up. We're at 21 up right now. There's a lot less snow. It was much more snowy when we were closer.
Right.
Unless the lights are just different. That's good. 990, 25 meters up.
Yeah. 990 meters.
Yeah, we're just going to stay at this depth until the end of the dive, just see what we have here. Do you want to go forward like a transect like we normally do?
Yeah, I think that's the best way to look for things to focus on. And then...
Oh, yeah, we're ready. We're ready whenever you guys are. Yeah, it's a mini transect. Ooh, that little bit ctenophore moved pretty well. Cool. He's swimming. Oh, how neat. They do look like little ghosts, don't they? We've been watching horror movies every night, so fits right in. Yeah, go for it. Let's see. I think it might just be schmutz, but I'll zoom past it.
Disappeared.
Yeah, it was out of focus.
Not out of focus, out of light.
Out of light, okay. This is a shrimp. That one looks different than the other ones we've been seeing.
Oh, that is definitely different.
That's like a yellow color. It's different shaped. It doesn't have the long tentacles. It looks like a candy corn.
Speaking of Halloween.
Yeah.
Oh, that's really gorgeous. So we are finishing up with our water column investigation for the day.
Yeah. Dive 3.
Dive 3. This largest of all biomes on Earth and least explored. Home to quadrillions of animals and...
Oop.
All right. Let's see what we got here.
Ooh, that's neat. It's an abandoned
Larvation house?
larvation house.
Exactly.
So now it's destined for sea snot. A little shrimp there. Oop.
Pushing out ahead. So we're only 15 meters above where we were.
Here's something.
Oh, yeah.
Ctenophore?
Nice. That's a ctenophore, a sidipid ctenophore.
Sweet.
Now, is that the same as we saw before or not? Sort of looks...
Longer?
Does it look larger? Yeah, exactly. All the buckets are full. That is beautiful. So you can see how it has the 2 long tentacles with all these side tentili, some of which are being pulled in right now, and then deployed again at the bottom right.
What is this?
Yeah, I don't know. What are those 3 white dots?
What is that?
Do not know.
That's weird.
Yeah.
Yeah.
That'd be great.
Oop.
Another
I've seen him.
ctenophore, yeah.
Yeah. It's these. He's missing out on his formation.
Yeah, exactly.
It's abandoned. Look at this crazy tentacles on this thing.
Yeah. Very impressive. You can... That's probably a pretty efficient way to hunt.
Yeah, I mean, you really can't. If you go anywhere near it, you're going to get snagged. Those are behind him. Those the white triangle. They're behind him.
It's very, very nice.
I have no idea what those are. They don't seem active. Like, I don't think they're alive. Maybe they are. 3 jellies, maybe?
Can't get a fix on what that might be. George Matsumoto thinks it's squid ink.
Squid ink.
Yes.
Yeah, it totally could be that. All right, well, there's another one at 9 if you want to get a good shot of that one. Seems similar.
Okay, that's great.
It's got to be. There's no
Ink?
formation of. Yeah, it's got to be ink. There's no body parts or. That's neat.
Yeah, I agree. That looks like squid ink.
Mm-hmm.
So squid ink as a distraction, hey, it works.
Yeah, distracted us.
It even confused us.
Stupid humans. Squid went the other way. So we're back up to 25 meters.
Mm-hmm. So it looks like that 10-meter mark is really where the.
There was a lot of action there.
A lot of biota in that depth.
Mm-hmm.
Had a little bit. There's a black fish at 9. Was it the big scale they called that?
Oh yeah, it does look like that same guy.
Mm-hmm. He very much mimics a squid, the way he moves.
Yeah, I like how it swims.
Yeah.
Interesting observation. Maybe he preys on the squid.
Yeah, he might prey on squid. I don't know. We'd have to ask shoreside that. You blend in with a group of squid and eat them. Yeah. Yep. Convergent evolution versus a camouflage. Oh, you're right. Maybe they do swim like him. Maybe he was first to the party. Yeah, what's that? Ctenophore. Little glowing ctenophore.
Nice.
Yeah.
Yeah, down at
They're common.
10 meters above the bottom.
You got an eel or something coming in? That's neat. Ooh. Excuse us.
That's nice. Yeah, you see the...
That looks like a gulper eel.
Gulper eel, yeah, that'd be cool.
Swimming like crazy, but that's what it looked like.
I feel like there were 3 red sidedipids down here. There's one that's red, like bright red, and there's this deep dark red, and the
like
Tinoceros one with the horns, the 4 horns.
Looks like a macaroni. Oh, who knows what that is? Poo.
Well, yeah, that's come from the water column above. And
Schmutz.
this is a great example of how the water column provides nutrients to the seafloor and to the benthic fauna on the bottom. It's all connected.
I know. It's like a giant food web or something.
Yes. It's like a one big ocean.
You got a fish at 1 o'clock, silvery thing. There we go. Oh, there you go. Ooh, I don't know what that is.
Nope.
It is a fish.
It is definitely a fish.
Nice. It's very shiny. Maybe photophores on the bottom?
Yeah,
see the little dots?
photophores and great eyes pointing up.
A viperfish?
Maybe a viperfish.
Sweet. He's pretty cool.
You have to get a little bit closer to the mouth.
Closer to the mouth?
I think it's a bristle mouth rather than a viper.
Bristle mouth?
Oh, so then it's just
Get up close.
There's his eye. I can see it really well now. The big black spot.
Cyclothony.
Oops.
Yes.
No, it's not a cyclothony. It's
What was that?
One of the big bristle mouths.
The large
It
bristle.
was one of the big bristle mouths.
Good to know. So this same family, different genus.
Ooh.
Oh, that's a nice little Medusa. I said, I think that's Ropolanema. Oh no, it only has 4 radial canals. What's that? Oh, I missed it.
Yep.
Yeah, I think it was a gonostoma or something like that.
Say that one more time, Mike.
Gonostoma. Gonostoma.
Thank you.
Or something related to it. What do I know? I'm not a fish person.
That.
Mike, at least
right
your
there.
things swim.
Woo!
Oh, I like that.
There we go.
That's a nice looking
Siphonophore.
Bison X siphonophore. Oh, it's just so beautiful. So I'm not sure what genus of siphonophore that was, but maybe we can get a screen grab and send that out to folks.
There's something at 9. It's red. Probably a shrimp. Yep. I've seen a few of these in bright red.
That's Lophogastrid again. Is that what that is?
I can get it to shrimp. That's about it. I can't even get it to a taxonomic name within the shrimps. It's
Yeah.
just a shrimp.
I gotcha. It is nice.
Maybe Penaeid? I don't know.
That was really neat.
Here comes something red at noon.
It's doing
Another shrimpy.
circles though.
Doing a dance. You're not going anywhere, man.
Do you think that's what we were just looking at
I
as
don't
well,
think
just from a
so.
different different angle?
Oh,
Or?
look how broken his back is. I don't think the other one had that back.
Mm. Great. Yeah, so this really is a different feel for it.
Mm-hmm. Another foraminifera, maybe? Oh, no, it's a lobate ctenophore.
So
We have a different fish at 9. And 2 shrimps. Yeah, that one looks a little funny. Yeah, he looks totally different than the other ones.
oh, that's great.
He looks really mad. There you go. Look how cool. He's
so
a big teeth.
cool.
I mean, big teeth comparatively. He's like the size of my pinky nail, I think. He's tiny.
Huh. I wonder what that is.
Ooh, he just opened his mouth. That was neat. He's scary looking. Ooh, ooh. I know, it's like the size of my pinky nail. But he's super neat. Staring up into the water column, waiting for food.
Yeah, I think someone will be able to identify him with
That was neat.
with that footage. Yeah.
Mm-hmm.
Yep, got that.
Ooh. Cadignath or something? Oh, maybe it's a fish. That's a fish.
That is a really cool looking fish.
It's very skinny.
Has a collar.
That's neat. Thank you.
Thank you very much. So, today we have been exploring, as we watch the siphonophore swim off, we have been exploring the water column, a little snippet of the largest biome on Earth, home to quadrillions of organisms, which is just a number too big to imagine. And these organisms that live in the water column have a huge impact on life on our planet. They're
A carbon sink, aren't they?
They are really important in the carbon cycle. They move carbon from shallower waters to deeper waters, and then also to the ocean floor, sequester that carbon.
They also feed basically the base of the entire food web.
Yes, they are excellent food for ocean sunfishes and whales and tunas and many different commercial fishes. And they also send nutrients to the bottom, where we're really interested in the organisms that live there as well.
Yeah, creating habitat.
Yes.
Feed the Lophelia reefs that we were seeing the other day.
Right.
It's a pretty cool place. Well, very, very limited exploration in this area. We're off of Florida on the Blake Plateau. Today I think our max depth was 1,000 something. 10?
Yeah, about that.
Yep, we did a couple of transects. We did one at 300, one at 500, one at 600, one at 700, one at 900, and then one 10 meters off the bottom, and then one 25 meters off the bottom. So we had a pretty productive day today.
Absolutely. And with all these transects, we also did environmental DNA sampling at 5 of the different depths.
Oh, 4, because one didn't fire.
Thank you.
So the fifth one didn't fire.
We were able to successfully collect eDNA samples for 4 of the depths that we had surveys for.
When we hit bottom, we grabbed 2 sea pens that were possibly new, or definitely different morphs.
Yes.
And we have a total of 11 samples, including our water, coming up.
Right.
4 of those are water.
And we had 5 organisms sampled from the midwater column. And they were a lobate ctenophore of unusual morphology that was not recognized as something that's known, a bright red ctenophore.
We have one with long tentacles, ctenophore with long tentacles.
Yeah. And then another red ctenophore that had little horns on it, had 4 horns on it. And then also a Narcomedusa, not Aegina, that was Bathychorus.
Bathychorus.
Yeah.
And then 2 sea pens. We are going to be having our pre-dive call for tomorrow at 4 o'clock. If you're on the line, please hang on. And if you want to join the call for tomorrow, just go ahead and call in. We're going to do that next before we process samples. So just hang out for a few minutes and we'll be with you shortly.
And thanks for joining us on this exploration of the midwater.
Yeah, hopefully we got more good weather tomorrow, and we'll be diving again tomorrow at 8:30. We should launch. So at the bottom around 9-ish. And we're doing the deep mound off the Blake Spur.
Exciting.
Or near the Blake Spur. Which we'll go over at 4 o'clock if you're on the line. So that will get broadcast.